Good morning, everyone. Can everyone hear me? Okay, great. For those of you who don't know me, I am Professor Ellen Pikitch. I'm Professor of Ocean Conservation Science here at Stony Brook University. And among the several things I do, I direct the Marine Conservation and Policy Program. And I'm here to welcome you and to convene our annual Marine Conservation and Policy Program annual symposium. So the students who've been working really hard for the past, most of them for 12 months, the program can be finished in 12 months, although some people by choice decide to take longer if they're part-time. And they've been working really hard, and they will be presenting their final project summary today. A warm welcome not only to the MCP students who are finishing, but to any new students who are here today entering the program. This is a great chance for you to see what you can accomplish in the program, the kinds of things, the diversity of projects that we have. And also I see there are a number of family members of students here, the cheering squad. Thank you for coming. It makes a big difference. It's great to have you here. So I'm going to ask the Dean of SoMAS, Dean Shepson, to come and say a few words of welcome.
Hi, good morning. Welcome. I'm Paul Shepson. I go by Shep. If you're students here, it's important that we have a great working relationship with all of the students. We're part of a team here in creating and providing, and with you having great experiences. So I mostly want to say you are incredibly important to us, you the students who are finishing and you the new students. This is sort of the main event, or one of the main events, where you show off and tell us about what you have learned in this MCP program. It's one of our most important, most visible programs. A lot of effort goes into making it good for you. I hope that's the case. The experiential learning part of this program is a key component. And I'm here because I love being here in this room for these presentations, because I'm always blown away by what you have achieved, how exciting it is and impressive your achievements are. And for those of you starting out, you will do the same. You will do amazing things in your career that we are sort of participating in helping you along with. So it is a very important program to us. Your success and your experience means everything to me and to SoMAS. I want to encourage you, if you have any feedback, there's probably plenty of mechanism for you to provide it to Professor Pikitch. But I like to hear from you. I like to hear accolades, and I like to hear complaints. It's more useful for me to hear complaints or constructive suggestions, because I can do something with that. Everybody likes to hear thank yous and expressions of goodwill and recounting your great experiences, but it helps me a lot if you have suggestions about how we can make the program even better. So you won't have as much fun as long as I'm standing here. So I'll stop, but thank you very much for being here, and congratulations to our students.
So Jessica Esposito is first. Jessica's talk is called Creating Resilient Coastal Communities with Project R.I.S.E. Her faculty mentor is Michael White, who is here in the back. And her internship supervisor is Alexa Annunziata from Peconic Baykeeper. Alexa is also one of our alumni from this program. So it's great that the program has been around for a while, and we see that not only do people finish and go on to get good jobs, but then they help other students along the way through internships and mentorship.
Good morning, everyone. Can you hear me?
Yeah.
Okay, perfect.
You don't need this mic.
Don't
You
need
got
that. Okay.
You got it right on. Yeah.
So my name is Jessica. I'm here to talk to you about creating resilient coastal communities with Project R.I.S.E. So some background, I work with Peconic Baykeeper this summer. Peconic Baykeeper is a great organization. They do a lot of work with Peconic Bay. But this summer we started Project R.I.S.E. And just a brief overview of their mission statement. They are dedicated to protecting and restoring Long Island's drinkable, swimmable, and fishable waters. And some background on the project. So Project R.I.S.E. stands for Recording Inundation Surrounding the Estuary. So we're looking at monitoring coastal change, specifically in the Peconic Bay, in and around the bay. And to do so, we use chronologues, which are a citizen science tool. I'll get more into that shortly. But this is a Peconic Estuary Partnership mini grant funded project. Like I said, it began in April and we have a 5-year timeline. So this project only just began, it's in its infancy, and it'll be exciting to see where it goes. So here's our study area, it's the Peconic Bay. We have 15 chronologue sites. Again, I'll get into that shortly, but why are we studying coastal change? Why does this matter? The National Oceanographic and Atmospheric Administration predicts that we will see about 2 to 3 feet of sea level rise on Long Island by the year 2100. And as we saw in that storm last, I guess Sunday, it was pretty devastating. And we can only expect to see more of that more frequently. So we're going to see an increase in storm intensity, an increase in storm frequency, a loss of habitat with sea level rise and coastal change. And that's especially damaging to the intertidal zone, which is important for species like horseshoe crabs and shorebirds, and also damage to property, livelihoods. And here's another, a bunch of other reasons why to study coastal change, but these are just a few. So what are chronologues and how do they work? So a chronologue is a post and we put it in the ground. It has a cradle on top and essentially you put your phone in that cradle and take a photo of the shoreline. So again, there's 15 stations and they all point to a shoreline. You take a photo, you capture the scene, you'll email that photo to chronologue and it gets uploaded into a time lapse online. And it's pretty instantaneous. And you can see how shorelines change day by day. Week by week, especially after storm events. It's a really important and useful tool and it doesn't really require much technology. You just need a cell phone to take a photo and you can email it whenever you want and it'll get uploaded. And the beauty of it is that these photos are in the public domain so that they can be used to advocate for policy change and lawmakers to kind of pay more attention to sea level rise and coastal change. And it also provides tangible evidence of coastal change. We get to see how these shorelines are changing throughout these photographs. So there's 15 current sites, 14 are active. We're still pending approval on one site, but it's really nice to connect with a bunch of different partners and hosts. So each chronologue is on a different piece of property owned by either Suffolk County Parks, New York State Parks, Peconic Land Trust, U.S. Fish and Wildlife Service, the list goes on, but it's really nice to see that everyone has the same mission of wanting to protect and monitor these coastal changes on shorelines. And each site is uniquely important. So it could be an environmental importance, like for shorebirds, recreationally important for places like Indian Island for kayakers and beach goers, or historically important, and they all exist in different habitats. So it could be an urban environment, it could be a salt marsh, a beach, and all of them have exhibited coastal change. So my objectives for the summer, one was public outreach, so I just wanted to get the word out about Project R.I.S.E. since again it just started. And I also wanted to increase chronologue engagement, so get people out there, get people taking photos of shorelines. And I also wanted to create interactive materials so that people can learn more about coastal change, why it matters, and how they can contribute. So what are we looking for when we take a chronologue photo? Wave action is just one example. We can see that bottom photo, the shoreline is looking a little rough. We have a lot of wave action, which increases erosion. And this is important, especially for a site like Elizabeth Morton, because there are New York State endangered shorebirds there, like the piping plover. And monitoring this erosion is important to protect their habitat moving forward. We also could monitor storm surge. This is Whittle's Hole in Greenport, and we can see on that bottom photo how much that water has come up. And this is a place where the dune has been compromised in the past. So again, monitoring these coastal changes and seeing how far that water is coming up can really help us better mitigate these issues. And also inundation. So this is Conch's Point in Southampton. As we know, we have daily tidal flows, we have variable tides, and this could just be an example of a variable tide, low tide, high tide. But it's important to keep track of these changes over time. And like I said, we have a 5-year timeline on this project. So it'll be really interesting to see how much these changes are evident in the next 5 years. So we did some outreach this summer. One of my favorite events was called Meet Us Out There. It was held back in June. We picked 3 chronologue sites. We visited them with members of the community, local government, and people from the private sector, and we just connected with the community. We told them about the history of the site, how they can use the chronologue, how they can get involved. And it was a really great turnout. It was really exciting to talk to people and hear their experiences with coastal change. And another event that we had was at Elizabeth Morton, and we talked about how these coastal changes might impact species like the Pikitch plover or other endangered threatened shorebirds. I also created a brochure. This is a super easy to use brochure. You can download it straight to your phone. And it gives you some project information, how to use a chronologue, but it also acts as a little adventure, so you can see what chronologue sites exist, where you can go. It has parking information, accessibility information, and it acts as a checklist, so you can kind of check off where you've been, where you want to go, and see if you can visit them all. I also created an ArcGIS story map using Esri. And it has a lot of project information on it. It has a really interactive feature. It's got a map where you can kind of scroll through all the sites, learn about their history, and it takes you right to the chronologue website, so you can view that time lapse and also learn more about the site owners, like New Suffolk Waterfront Fund. And it's a really fun feature, and it kind of gets you to scroll around and see what is out there. So what were our results from this summer? So we're mainly just tracking engagement, so we'll be tracking captures and contributors. So captures are the photos, contributors are people that took the photos. So in the last 30 days, we've had over 50 photos from 40 unique contributors, which is really exciting. But the most exciting thing is that as of yesterday, we hit 200 total photos. And we've had a 50% increase in these photos from May to July, which is my internship time. And just seeing that increase is really special because we're getting more people out there, we're getting more people to learn about coastal change, how to get involved. And we've had over 130 different people get involved so far. So it's pretty exciting that since April, we've had over 200 photos. And our most popular site so far is Elizabeth Morton National Wildlife Refuge. It's pretty accessible. As soon as you get to the end of the beach trail, the chronologue is there on the observation deck. And it'll be interesting to see how this site manifests over time with the coastal change and chronologue photos, since we're getting so much engagement, so much activity. So what is the importance of citizen scientists here? The value of communities. Each person has a really unique relationship with the coast on Long Island, whether it's the Atlantic Ocean, the Sound, the Bay. Everyone has a relationship with the water here, and it'll be really important to get people out there and involved with coastal change moving forward. And this project would not have been possible without the community and their support. Like I said, over 130 people have showed up to take photos of the coastlines and monitor these changes. So moving forward, it'll be really important to see how that manifests itself in the next 5 years. So some of my recommendations for the project would just be to continue outreach. The more events, the better. Just getting people out in the water to see the coastlines, especially after storms, is really beneficial. I also think having some more signage for these sites would be great to kind of bring people in and see that there is a chronologue there and how, what they can do with it, how it works. And I also think more sites would be great. This is in the works, but I think it'd be great to involve more communities and get more people involved in coastal change in and around the Peconic Bay, especially environmental justice areas moving forward. I think just getting a different diversity of groups of people involved would be great. And in the future, we could consider future flood level analysis. So there's a program called RCNN. It's a spatial analysis program. It's probably very costly and time consuming to analyze, but you actually can use it to take a look at objects in a photograph and see how much the sea level rise or the tidal flow is changing in photographs. So it's pretty interesting, but that could be just something in the future for data analysis. And also connecting the community through storytelling, whether that be a blog or outreach events. I think it'd be really great to have people from all different walks of life get involved and talk about their experiences with sea level rise, with tidal changes, storms. Like I said, that storm at Stony Brook last week was very damaging. And I just think something like that could be really beneficial for this project, and just having a blog where people can talk about their experiences with storms and, moving forward, what they think of coastal change. So I'm excited. And I want to thank Alexa, Pete, and Vicky, and Michael White, my advisor, for a great summer. And now it's your turn to go ahead and capture and contribute. So if you're ever out in the Peconic Bay, on the left, I have a QR code for the brochure, and on the right is the interactive map. So feel free to scan either of those. And if anyone's taking classes at Stony Brook, Southampton, Conscious Point is just a few minutes away. So if you want to make a stop there, it'd be really awesome to help contribute to coastal science.
Great. Yeah, great job and great project. And not only that, your enthusiasm for the project really comes through when you present it. Anyone have a question?
So, a lot of the stuff I've done is with tides, so it always gets me curious. It would be really cool, and you may already do it in your like ArcGIS or when you're showing like those chronologues to be able to say whether it's high or low tide.
Yeah.
And does the data, so, and then I guess the question is, have you at all, or anybody in PBK started like trying to correlate those pictures with the tide levels?
Not yet, but that is something that could definitely be looked into. Right now, it's just more of getting people out there and just
Yeah.
starting like the project. But I do think that's a really great idea, and that'd be really interesting to see too, especially like marking if there was a storm or something that moved through and just correlating those two things. I think that'd be really interesting.
We determine property like boundaries along the shore by mean high water level. So it'd be really curious to see if high water is changing over time.
Sure. Yeah.
Good point. Other questions?
Yeah.
One way in the back there.
Perfect. You mentioned that sometimes these, time-lapse photographs, could help with contextualizing, policy agendas or policy maker usage or something like that. Do you have any examples of that, occurring or in your project or in others that are similar to this one?
I don't know if that has led to it, but I grew up in Massey Beach in Shirley, and I've just seen firsthand that people are always putting photos up on Facebook of storm damage and tidal damage, and it's really helped get people involved. And I know Dan Panico and other local officials for Shirley and Mastic have gotten involved based on storm damage, and I just think that this could be a really great opportunity to use these photographs for something like that. And if there is damages from coastal change, I think it'd be a really great step to use that for something like that.
Just curious, do you have before and after pictures, Sunday and Monday morning?
These were a couple, so this was
Wow.
one example of storm surge at Whittles Bowling Green Port and Elizabeth Morton, so you can kind of see more of the wave action here. But again, this project just started, so some sites have more activity than others. But for the most part, I thought this one was most interesting, especially just how far that water's come up and knowing that the dune was compromised at this point at this beach at one point in time. I just think it's important to see those before and afters. So it'll be exciting to see more once we get more photos.
Any more questions? I've got a question.
Yeah.
So it seems like, the success of this really depends on having lots of citizen scientists willing to participate, but also, you want to have good coverage of the sites at different times. So do you ask the citizen scientists to commit to going to a particular site on a regular basis, or do you just, whatever they do, they do?
Whatever they do for the
Okay.
most part. I mean, we do have more sites that are active than others. Downtown Riverhead is a pretty accessible site, so that one has a lot of activity.
Mhm.
Elizabeth Morton is another site, and then some sites like Shelter Island are just kind of a little further out of the way. So if someone's hiking on the trail and happens to come across the chronologue, they can take the photo. But really just depends on the community and their willingness to go out and take a photo.
So do you see any, is that something you would consider changing? At least having one component of the citizen scientists who would go regularly and commit to.
Yeah, I mean, I think it'd be interesting to kind of have like an ambassador for that area, kind of take charge and get out there and regularly capture the sites.
Great. Any other questions come to mind?
Yeah.
We have one here.
Just following up. Did you notice through your research, were there other sites like this? I love the idea of citizen scientists. Are there other sites like this across the country, like to create awareness? I think this is excellent.
So chronologue does exist around the country. I know a lot of national parks use it, just to kind of capture what people are seeing. I don't know if it's used in this capacity, but I do think it'd be really cool if this was expanded all across Long Island to get all communities involved, not just the Peconic Bay. Even Mastic and Shirley, like I said, that's going to be a very problematic area moving forward with coastal change and sea level rise. It's a very low-lying area, and I think it'd be really cool to have something like that in different communities to get everyone involved. So hopefully that's something that can happen in the future.
Any more questions?
We're at 14 and a half minutes.
Oh, okay. All right, you're out of
You want-
time. Thank you very much. Let's have a big round of applause. So our next speaker, I've gotten to know pretty well because I was her mentor in her capstone project, Silvia Gao. And the title that she picked for this project is Technology Advancement Options for Shinnecock Bay Restoration Program. Good to see you, Sylvia.
Good to see you too.
Okay. Take care.
Okay, good morning everyone. So my name is Silvia Gao. My project, as Ellen already mentioned, I work with Shinnecock Bay Restoration Program, or for sure, SHRP, this summer, and trying to understand what are the options for technology that we can use in the future. So if you guys know me well enough, you know that I used to have a career in software and IT. So this is actually a very relevant project for me and also just for my past experience as well. But fun stuff first. So one of the benefits of working for Pikitch Lab is that I can personally join all of the very amazing boat trolls. So we usually leave from Southampton Marine Station, and usually we survey about 11 stations every time we go out. And I help with collecting like water quality survey and data collection, and also helping with pulling your trolls, counting crabs, and it's just really cool to touch some animals on the way. But on top of that, serious information here. So actually, a few months ago on June, we had a kickoff for the World Ocean Week at the Explorers Club. And Professor Pikitch, she was there personally to receive the flag from the Explorer Club. Because we actually have a very exciting expedition coming up next month. And because of the high profile and there's a lot of innovation and technology we're using in next month's project, we were able to have this amazing honor to receive this amazing flag, which has been to a lot of places like highest mountains or deepest ocean and things like that. So it's definitely a very cool recognition for us to have, not just from the research field or scientific field of everything, but also from the other organizations. So that is very exciting, even though I wasn't there. So one of the cool things about the expedition is that we're gonna use this open ocean robotic data explorer. So as you can see, this is basically a boat that can be outside of the ocean without any humans stationed on the boat. So it could be out from weeks to months in very choppy, very dangerous waters, collecting data and also samples without a lot of human interventions. It's gonna save a lot of money, and we are gonna be one of the examples showcasing what we can do with the amazing technology. So as you can see, this boat is basically as tall as a human. And we have a lot of like cameras and sensors that are already pre-installed. We can also add a lot of other integrations on top of what we have already as well. So with that, we can also leverage the functionality of like auto correction, or we can also set the route when we even before we set the boat on the ocean. So again, a lot of things that we can do. We can also basically manually intervene while the boat is out, so that we can correct where do we want to go with the boat as well. And not only we're using this very cool boat, we're also leveraging a new set of eDNA sampler technology as well to be integrated with the boat. Yeah, okay. So to be integrated with the boat, basically nowadays what we're doing right now with the eDNA sampling is that we go out, we go on the shore, we collect all of the water samples, and then we have to send it out to the lab on the same day for a faster processing. Another thing is, if we're sending out a boat for like 10 or even 15 days on the water, what are we gonna do with the samplings? So the answer is that this company, McLean, they have those kind of sampler technology that we can have up to 48 filters on the vehicle. They're gonna automatically collect samples and also preserve them, so that after the entire expedition, and then we can send them all together to the lab for analyzing and also for future analysis. So anyways, I think it's very important to share with everybody what we're doing here. Even though the expedition is going to be next month, and I won't be personally here, very sad. But I thought it's really important to know that those kind of very cutting edge, amazing technology is not that far from us. We're actually doing it here at Stony Brook University under our department, and it's really, really inspiring to know that this is actually doable and it's not that far from us. Now, let's talk about what I actually did. So basically last year at the UN meeting, Professor Pikitch received 2 of the donations from technology vendors, and Professor told me, how about you take them home and try to figure out how to use them? So I thought, yeah, sure, why not? So one of the 2 is this spotter device. It is a flexible solution for real-time surface and subsurface sensing. It has native sensor only for temperature, wave, pressure. We can also add more sensors on top. As you can see, it is solar powered, don't have to charge it that much. It can be out on the sea, floating around or more in a different way to collect data. And you can also have the data accessible online with the dashboard. So yeah, I thought that is really exciting. So I really want to figure out how to use it. And one thing we also learned that it's a very portable device, at least according to their website. You can carry it with one hand. So I thought, it's gonna be easy. Not really. That's what happened when I picked it up from the marine station. What I didn't know is that we have the spotter. We also have other things attached to it. So what you can see on top of the yellow part is that this is the smart mooring system here. So you have sensors here. You also have sensors here, here, and also here. So I definitely forgot how much that thing is gonna take in the back of my car. But again, you can also know that with this kind of design, we can flexibly adjust how deep do we want to collect data with what kind of sensor that we want to attach. Another thing that we can do, but we didn't pay for that, is a integration system. So with the Bristol mount, customers can also add like more sensors to collect more data here, including even connecting with cameras or hydrophones. So again, it's a very, very flexible open source solution that also costs like $3,000 more money. So we didn't do that part. But anyways, I was able to restart the device and also retrieve the data from 2023 when we were testing out in the marine station. So as you can see, you can have like the wave information and more wave information as well, and also temperature. Another cool thing about this vendor is that a lot of other customers are also sharing whatever their data that was collected by the spotter, and we can go to their online portal to look at their data information as well. So even when we have questions in the future, and if we don't have any devices, we can still go there and find out what's happening over there. So that's what I did with the spotter device. Another part that we had was a whole new tidal gauge. It is also a donation from last year, and it's a real-time solution for us to monitor water level and also tidal activities. It is also solar powered, so it can be just easily stationed and left there for a very long time. We can also access data online very easily as well. And most importantly, this one is actually portable. The size of that is basically like this. So it's really small and very easy to deploy. Only if it is not broken. So this is what happened when I have to manually reset the device, and you can see the inside of the machine is basically just rotten from leakage and also too much moisture from the marine station. So that's the end of the story of the actual portable device. Another thing that I kind of studied, but we didn't really execute on the deployment of the project, is all of the AI related image and also video processing. So this is actually an image from one of the vendors converting pixels to impactful insights. So one of the companies called Ocean Vision AI, they are actually invested by NSF and Endeavour as well. So they were trying to develop software to help us analyze images and also videos by using artificial intelligence, machine learning, and also crowdsourcing to help us better analyze and identify objects in the deep sea. So as you can see, they're working with Endeavour, so they definitely focus a bit more on the deep sea rather than near shore or coral reef kind of ecosystem. Another company is Sea Vision AI. I'm not gonna spend too much time on those videos, but one of the examples I want to share is that one of their use case in New England. I don't think the video was playing, regardlessly. What they were trying to do is that they can basically use the software to count the amount of flounders that were swimming through the net. So that was also something that we were trying to do in Shinnecock Bay restoration program as well, because we also have years and years of video and image footages, and we were trying to figure out if there's a better way of analyzing what we have over there. But one of the recommendations over here is that we talked to the vendors, and they were even happy to do a 1-month trial project with us to deploy and test out the algorithms and tailor it to our requirements. But what they need is actually a very specific and also solid use case. Like for example, where exactly are we stationing the cameras? Is it going to be in the middle of the water column or in the bottom? what are you actually trying to identify? Is it fish, crabs, or marine debris? What do you want to do when the system can detect something? Do you want a student to come over and manually note down the name of the fish, or do you want the system to automatically count numbers? So until we figure out all of the questions, it is basically a waste of time and energy to invest in those kind of projects, and we have to answer those questions within our organization first. The other few things I would recommend is, I do believe the SoFar starter, even though it's a bit chunky, it is gonna be great to actually deploy that in the ocean, especially for any station that is, more curious to us. And then tidal gauge is on a pause because of the cost, and also because we don't have a very urgent need for tidal and also water level monitoring. So yeah, that's a summary of my capstone project. I'm ready for questions.
Great. Thanks, Sylvia. Any questions?
I don't have a, can you guys
comment.
hear me? Oh,
All extremely interesting and exciting, but those things look to me like they would be very dangerous to navigation and easy to simply hit with another boat driving at night, which is what I do very frequently. And they certainly should have radar reflectors on them at the very least, because they're really low and would not be very visible in the dark. That's my suggestion.
Right, it's actually a good question. So actually for the boat part, I do believe that when they're actually talk about something like their auto detection system, so we didn't really look into all of the details, but I do believe like they can kind of detect boats coming over, or if you're in a very shallow area with a lot of rocks and seaweed, they will be able to like detect and alarm whoever that is on shore, so that we can kind of detour around as well. With the spotter, I do agree it is like a small device. It only has a small light on top. I do think once we actually use that in real life, we need to at least put some other things to make sure people can see it. But also another thing is that I do believe before we put anything out in the open water, we need to have at least some kind of approval or like discussion with the local whatever government it is, to make sure that we're not in the middle of the route of any boats or a lot of like swimmers around. So there are definitely things we need to do for that as well. Yeah.
Anna, here has a
Yes.
question. Yes.
can you hear me?
Yes.
All right, cool. Well, great job, Sylvia. That was an awesome presentation. I just wanted to say actually my internship, where the place I was interning at, got one when I was there, like in the middle of interning with them. And it was really great because in the morning, like we would just get data of like the visibility and the temperature and everything. And so it just made our dive, like we were more prepared for our dives. And we did, because like there's like that red buoy that's connected to it, like we just wrote all over it, like don't touch, don't move. But it was in a pretty shallow area, so it wasn't really sorry, I'm just the Simpson thing is cracking me up and it's like flashing and I'm like it's like getting my attention. Sorry, thank you. It's also like on the screen behind you, so I was like confused. But anyway, I just wanted to say that it was it's definitely like a cool thing. So I was like, oh my God, wait, this is the exact thing you were talking about. So we used it and it was great. So highly recommend. Yeah. That's not really a question, but just being like, I was really excited to see that you were talking about it.
Yeah, I agree, because I feel like in the future we're gonna be sitting coffee in Swenson room over there, and then we will be able to understand like what's happening out there very easily to plan our day or our patrol. So definitely gonna save a lot of hassle for a lot of us.
Other questions for Sylvia? Go ahead.
Yeah, so I'm a little unclear what the hopes and dreams are for you or the team in terms of data that you will be able to obtain with this automated sampling that you are not able to get with human, from drawing. What can you do in your view that you are not able to do now?
Sure, great question. I think one thing is that we will be able to get access to very difficult water areas. like for example, extremely choppy or very shallow area where our current troll boat might not be able to access to. I think we will be able to collect very precious data from over there. The other thing is that I think most of the trolls have to be operated in a very specific time of the day. We're usually out during the morning all the way until 1 p.m. But it's gonna be very hard to collect any information during the nighttime. But how about the fish that only comes out during nighttime? We can't really like do that much with the troll. So now with those kind of samplers, we can collect data from day to night all the time, even including when it's raining with a storm or just very cold winter time when we cannot go out to troll. The other thing is that the boat is gonna be more quiet than our current troll and is gonna be less destructive from for the ecosystem as well. So I do think that's also gonna be another improvement. Yeah, and also currently the troll cannot really go out that far because it's basically a half-day trip. But once we have the availability of a more powerful and capable boat, we can basically go a bit further away from places that we cannot explore right now. Hope that makes sense.
It's great.
That's fine.
That's it?
All right, thank
Okay,
you.
thank you. Yeah, let me just add that this expedition that's going to happen starting on the 9th of September in Shinnecock Bay, it's not only to assess the biodiversity through eDNA in Shinnecock Bay, but it's also the first time ever that an autonomous surface vehicle and an eDNA sampler have been integrated and are working together. And so it's proof of concept of the technology that we're trying to show as well. And you know, my dream is that these kinds of integrated systems can go out to remote parts of the ocean because the device is solar powered and can stay out for 6 months at a time, can withstand hurricane force winds, is self-righting, that we can be doing biodiversity surveys in the remotest parts of the ocean without having to go out and refuel them and things like that. In terms of helping us in Shinnecock Bay, you know, as you know, Dean Shepson, as well as anybody, we have a limited number of research vessels available at Southampton, and the demand for those vessels is enormous. And so when I planned my research, I've got to ask, you know, Brian Gagliardi always asks me way in advance, what days are you going to want this boat? And if something happens and we need to change, it's often very hard to do it. So this is a way to get those samples without needing one of the research vessels, without needing a crew and a captain. Usually we have at least 10 people on the boat. So it's, I think it's just going to make it much easier for us to be able to do the sampling we need to do, and also to encourage people around the world to think about non-invasive, non-destructive solar powered technologies. Anyway, sorry to take some time on that, but I'm excited about this expedition. It's been a long time in coming. All right, our next speaker is also one of my capstone students, Serena Bandlow. Serena, come on up. And the title of her talk is An Examination of the Spatial Distribution of Elasmobranchs in Shinnecock Bay.
So how about that? Dr. Pikitch gets a double header. So yeah, for my project, I looked at the spatial distribution of Elasmobranchs in Shinnecock Bay. So this is in a lot of ways very similar to Silvia's stuff. I was also working with the Shinnecock Bay Restoration Program, SHRP. It's a big multidisciplinary program that was launched about a decade and a half ago to address the ongoing ecological decline in Shinnecock Bay. And namely, especially in the Western Bay, which was in really bad condition at the start of this, constant brown tides and whatnot. And a lot of the focus of it has been improving water quality, restoring seagrass beds and oyster reefs, and it's had a lot of success over the years, to the point of even receiving recognition internationally as a Mission Blue Hope Spot. I also just made a quick little map in GIS to show where it is. So some of the motivation and objectives of this was to understand the spatial distribution of Elasmobranchs in Shinnecock Bay, in part just because I think shark skates and rays are neat, and also because they're important ecosystem constituents if you want the boring science answer. So of course, a part of this is comparing the distribution and presence in the eastern and western regions of the bay. Identify what might be influencing this distribution and just generally contribute to the ongoing efforts of monitoring and inform future strategies of conservation. So one of the main methodologies that was used here was environmental DNA, eDNA, which we could collect from the water just simply by taking a bottle and scooping up a sample from the water. It's stuff like excretions, scales, sloughed skin. It's really nice because it allows for detection of species presence without any direct observation or capture. Doesn't do any harm, doesn't injure anything. And it's also very useful for rare and highly mobile species that the trawls that Silvia and I participated in sometimes miss. And it works kind of as like a snapshot of what's there roughly around that time. So as I said, the main method was the eDNA water sampling, which was done monthly in July, August, and is being done in September. Due to how long it takes to process it, the data that I will be showing today is actually from last season, but I did participate in the collection of it this season. Once the eDNA is collected, we bring it back to a lab here at Stony Brook, where we do extractions and amplifications before sending it out for sequencing. And what I did with this data was I created a number of maps in ArcGIS Pro to visualize species distribution and richness. I also ran a few statistical analyses just to try and more numerically confirm what you can see visually here. So let's run through some of these maps. There's a lot, so I'll try and have some brevity here. So smooth dogfish was very interesting to me because this was the only species that we saw at every single site, every single month. So you see times detected 3 for all of them. There were some that we didn't actually get to sequence all the way down to the species level. For these, we saw them a little bit more varied. I did run some statistical analyses to see if this difference is significant, and it's not. But it does look like there's a little bit more presence in the East Bay than the West Bay, just at a glance. Although, of course, part of that's informed by there being more sampling sites in the East Bay. And those are really the only 2 species that had truly robust data sets. Basically, everything else was detected 5 or less times. So clear nose skate, 4 times in the east, once in the west. Bullnose ray, very similar story. Cownose ray only in the East Bay. Spiny butterfly ray was again only in the East Bay. Same for the only rhinoptera that we saw. I'm sorry, I'm gonna butcher that pronunciation, guys. Rough-tailed stingray was the only one that was only detected in the West Bay, which I found interesting, but again, it's just one data point. And little skate and sand tiger shark were both only seen once each in the East Bay. So these were some of the more interesting figures that I think I have here, because these are some of the ones that show some statistical significance and some stuff at least approaching it in some cases. So for species richness, well, to me, this looks like it's approaching statistical significance here. It actually doesn't quite reach it. However, the dissolved oxygen for the months of August and September does reach statistical significance, and I'll show that in some tables in a moment. But it's very interesting that the less healthy part of the bay had a harder time maintaining high enough oxygen levels throughout the season. And I can say that anecdotally, we saw the same thing this year on the trawl, not the most recent, but the one before that. We actually, here at the Dune Road site, saw DO of flag 3, which, yeah, we did not pull up much living in that trawl. So here's some of the t-tests that I ran. You kind of have to take the numerical counts with a grain of salt due to the difference in number of sites. But you can see, well, the total detections, the richness does get a little close to statistical significance. It doesn't quite reach it. You have that again with temperature in September, which also gets close but doesn't quite reach it. But salinity never reached it, temperature never reached it, but in the later months of the summer, the DO does reach it. P-values of 0.017 and 0.001, which, yeah. So, yeah, there was definitely, at least visually, an observable increase in elasmobranch detections in the Eastern Bay in August and September when compared to the West, though it did not reach significance. Smooth dogfish was a very interesting outlier to basically all of the other data, because it's just the one that's everywhere. And I can say on the trawls this year, that is by far the most common elasmobranch we found. Clear nose skate was the only thing that came close. There was generally a preference for the Eastern Bay among the individual species, and I imagine if you took out the dogfish data, it would much more heavily look to favor the East Bay. And the only real significant differences that were found were this crash in the DO in the West Bay in the later months of the summer that was not matched in the East. So overall, I would say that the results indicate that there's been some success in improving the overall bay health. There were not significant differences in richness or species distribution. There's definitely an issue with the DO that needs to be addressed, and perhaps that could be done with collaboration with water quality data from other labs, an increase in sampling frequency and eDNA to see whether we're just maybe sampling at the wrong moment. I know talking about this glider expedition, it'd be really neat to get to see what's there at night, or even just other times of the year than just the summer. And it'd also be very interesting to incorporate some more environmental parameters, such as, well, maybe they're here because their prey's here. Maybe they're here because they prefer the habitat, or their predators aren't here, or they prefer the depth of the water. But yeah, so in conclusion, there really were not any significant differences in the distribution or richness of elasmobranchs. But there were some in DO which indicate that the West Bay is still in worse condition on some level, but that this is a valuable methodology for continuing to monitor marine ecosystems, and, well, there's always more work to do. So here's my references. I'd like to thank Dr. Pikitch. I'd like to thank Libby Salzman and Natalia. Hi. And also my parents. Hi. Thank you. Any questions?
Many openings to the ocean does the bay have, and what divides East and West Bay? Sorry.
So part of the bay is kind of cut off in these figures, but on this, it's just the one inlet here is the big one. And you can see there's a big narrowing. There's a bridge right here.
Concord Bridge.
Yeah.
So I'm just thinking that maybe you would see more sharks in the East Bay just because the ocean, the opening to the ocean is there.
Yeah.
So the oxygen.
It's entirely possible. That's a big part of it.
Okay,
Yeah.
good question.
What do you know about how long the DNA lasts in those environments, and how does that affect the quality of the temporal information that you can get? When are these species there, and how long ago might it have been?
Yeah, that's a great question. It doesn't last long, isn't it? Just like about 24 hours-ish. Yeah. Obviously, different types of things can persist for a little bit longer, but generally, it's basically just a snapshot of what was there in the last couple of days at most. And of course, you're right that can influence things. Like we went out on a trawl this last Monday in the midst, like just after, before even, a big storm. So that can affect everything from the amount of oxygen in the water to the salinity, which could impact what's living in a specific area. So that's why, obviously, finances aside, it would be great to be able to do this more frequently.
This must mean you have to do the amplification right away.
Oh yeah, you've got to run it right back to the lab and do that stuff within like the next couple of days.
And I want to acknowledge that we are happy to be working in Gordon Taylor's lab. Thank you, Gordon.
Thank you.
Without Gordon's help and use of his lab, this wouldn't be possible. So we're rushing the samples to him in the lab and doing the work. Other questions? Caitlin.
Yeah, is this, so this eDNA research has been done just for the last two seasons, or it's much longer?
Well,
So
I'll let you, do you want me to answer? Or you
Yeah,
know
you
the
can
answer?
answer that.
Okay, so this is the 5th year that we've been looking at eDNA, but it's the 3rd year that we looked at Elasmobranchs.
Gotcha.
Mhm.
And it's a good question, so Serena, I was gonna, if I can riff off your question. When you found no statistically significant differences, you only looked at the data from 2023.
Yes.
But we're going to have data from 2024 for
Mhm.
Elasmobranchs, and we do already have data from 2021. So I have a feeling that some of these patterns will be statistically significant.
Oh, yeah, it'd be
Yeah.
very interesting to do something basically just like this figure, the monthly species richness, but yearly instead. That would be very interesting, I think. Doing it for
By year and
individual
area.
species would be very interesting too. It's what's so nice about this ArcGIS stuff is that once you get the map set up, the framework, it's honestly kind of plug and play. So it's very easy to just be like, okay, now I'm gonna throw the DO into the same framework or whatnot. You had a question?
So the West Bay is very shallow, so
Yeah.
what's stealing all the oxygen?
Honestly, a little bit beyond the scope of what I looked at here, but that is the big question. It might just be that, like was said, there's not an exchange with the ocean that makes it a little bit worse. It could just be phytoplankton blooms. I think we did have a big bloom at one point when we were going out there. But yeah, can't say with confidence for that.
It's fertilizer.
That would do it, yeah.
It's
Runoff and
it's
fertilizer.
temperature also. I mean, well, anyway,
Mhm.
I don't want to answer your questions. You're supposed to answer the question. I have a question if no one else does. So you showed a lot of data.
Yeah.
Can you talk a little bit about how the number of Elasmobranchs that we've seen using eDNA compares with the number of Elasmobranchs we've seen using just the trawl, the catches in the trawls?
Yeah, well, really, the only thing we've seen, we've had 1 or 2 rays. I did not get the species ID on that. We've had like maybe 5 clear nose skates total and maybe a similar, slightly higher number of smooth dogfish. And that's really about it. So most of the species here, like we certainly never pulled up a sand tiger shark. I don't even know if we could fit one of those in our net, but so it's part of the value of this is that it lets you see these species that you would miss otherwise with the survey trawls. Obviously, it alone isn't maybe enough because it's not showing the scale. It's not saying, okay, we found 2 dogfish and 3 clear nose, but it does tell you, okay, well, we're seeing clear nose in the inlet and South Grass, which I think is very valuable.
That's it.
That's it.
Awesome.
You're out of time. Thank you very much. Good job. We'll stay in touch.
Absolutely. Hey, we need another trawl puller next summer.
Okay, great. All right, our next speaker is Cassidy Brown.
So Cassidy's is actually recording today.
Oh, okay. So we're gonna watch a recording.
Cassidy, we're going to watch a recorded presentation. She is, whoops.
Okay, and while she's getting ready, her faculty mentor is David Taylor. David can't be here today, so he's on Zoom.
Yes, he is.
And our internship supervisor is with, I see that Rob is here, but is Ellie here? There you are. Ellie, I'm not sure how to pronounce your last name. Seiler from AMCS, which is Atlantic Marine Conservation. Oh, I always get this wrong.
Atlantic Marine Conservation Society.
Right. And AMCS has been a great partner with the MCP program pretty much every year. They've taken on interns and in some years more than one, and students always learn a lot and have a lot of fun. So thank you.
So I see that Cassidy made this a little bit long,
Okay,
but I'm going to start it and then let me know if the volume's not good.
My name is Cassidy Brown. I'm sorry I couldn't be there in person today. But this summer I was lucky enough to intern with AMCs under the mentorship of Professor David Taylor. And what I chose to focus my research project on was marine debris analysis of Long Island beaches. So while interning at AMCs, I don't know if everyone's familiar with AMCs, so just like a little bit of background there. They are the Atlantic Marine Conservation Society, located in Southampton Beach. Their mission is to promote conservation through action. So how do they do that? Primarily, they respond to stranded marine mammals and sea turtles. They also rehabilitate cold sun Kemp's Ridley sea turtles every winter. They participate in community outreach events and host their own educational programs. And they perform their own wild population studies, especially of seals. So just to break down those different actions that they perform a little bit more. Stranding response, what is it? Like, what is a stranded animal? In general, the definition for a stranding is a sick, injured, or deceased animal that washes up on our beaches or our shores. For AMCs particularly, a stranding is a deceased, dead, marine mammal or sea turtle, with the exception of large whales. So AMCs will respond to large whales if they are alive or dead. So these animals are dead, washing up on the beach. So what is response to a dead animal? Like, what are we doing? Well, when we go and respond to these animals, we perform a necropsy. A necropsy is just an animal autopsy, and the purpose of doing this is to investigate their cause of death. If we can determine that, we add it to the NOAA database and share that information with all of the surrounding network. And we also investigate possible human impacts, which is a really huge part of what AMC was doing. Whether that human impact be vessel strike, entanglement scars, marine debris ingestion. We want to know if that, like us as human anthropogenic influence, is causing death to these animals, or maybe if it didn't contribute to death, it had an impact on their life, and we definitely want to document that. Oh, sorry, I jumped ahead, but like warning, this next slide is a little like gross. I try to choose the most friendly photos of me in action doing this summer, so if you are Spanish, I would just look away for a quick second. So performing these field necropsies, this is a lot of what I did this summer. It was a lot of field necropsies of many different animals. My first day I did a harbor porpoise and a harbor seal in one day. I've done probably like 5 loggerhead sea turtles, 3 dolphins, 1 minke whale, a bunch of Kemp's Ridley sea turtles, a green sea turtle. So my summer was filled cutting open dead animals, which was more than I thought I was going to do because I was hired as an educational intern. So I definitely got the hands-on experience, and that was really cool. As you can see, in the top left, that's me, and bottom right, that's me measuring a white-sided dolphin that died near Ashroken Beach in Northport Harbor. And in the middle is a minke whale that we responded to in Rhode Island. The fact about that experience is that I got to cut the heart and lungs out of this animal with a very large knife. So it was sad, but also kind of cool. Definitely wasn't expecting to be able to do that this summer. And in the bottom middle, that's me and a woman from Mystic Aquarium going through the intestinal tract of that same whale, looking for possible marine debris in its intestinal tract, or any like cysts, parasites, abrasions, like anything we want to document for this animal's death. And then on the top right, that is a loggerhead sea turtle that was hit by a boat near Port Washington, washed ashore, and I performed a necropsy on that animal as well. So a lot of that this summer, and that's a big part of what ANSES does. It's very important work, and the documentation that they're getting is really going to help conservation measures in the long run. So next, they also do sea turtle rehabilitation. So this winter, they received 20 cold sun chemistry sea turtles from the New England Aquarium. So cold sun just means hypothermic, and Massachusetts gets a lot of these cold stunned animals every year. So we help them out by taking them from overburdened facilities and rehabilitating them over the winter and releasing them. So we received 20 back in December, 16 were flown out. In April, they were flown down to Georgia where the water was warm for release. And then the last 4, I got to help out over the summer with husbandry, including feeding, taking measurements, checking the water quality of their tanks, performing any medical attention they might need. But those last 4 were released at the end of July. So I also got to participate in the sea turtle releases, which was really fun and cool because you get to involve the community, answer questions while all celebrating a really positive event. And a fun fact, all of the 20 sea turtles this winter were pasta named. So we got to release like orzo, cavatappi, angel, gemelli, just like fun little things like that. Yeah, that's on the left-hand side gonna be me taking voucher photos of orzo right before his release. So we do a lot of documentation. That's orzo and his banana box ready to go. And then top right is gonna be him entering the water. So in addition to that, they do wild population studies. So ANSES is doing seal surveys, whether that be aerial, boat, land. And these are important because we get to assess the population size of these seals, where they're hauling out, so their distribution and overall habitat use. In addition to the surveys, they are performing seal captures, so that is just restraining a seal, taking biological samples such as blood and skin and blubber. And we use those samples to assess if there's like a virus going around the population, if there's anything abnormal, just to be an indicator for the overall population, anything that we need to worry about, since we do live in proximity to these animals. And then we will put a satellite tag on that seal. And that way we can track movement patterns and then also like get a better idea of distribution, habitat use, where they're going, where they're feeding, etc. And then also for our cold stun sea turtles, when we release them, we do put tags on them, whether that be a satellite tag, if they're big enough, an acoustic tag, and they all get pit tags, which are basically like the same as a microchip in your pet. And these help us track their movements, how that changes through time, where they're going, and how they're using like our Long Island waters when they come here. And this is me helping our chief scientist, Robert DiGiovanni, put a acoustic tag on orzo before his release. So that's the parking lot of the beach, and little guy is getting an acoustic tag, so we get to see if he comes back, when he comes back, and where. So the key part of what ANSES does is outreach and education. So this summer I got to go into a lot of classrooms before school was out, teaching kids about the wildlife that we have here, how they can help, what like the wildlife needs from them, and just like increasing awareness overall. We got to do teaching necropsies, so teaching the older kids a little bit about turtle anatomy. That was cool to see their reaction to that, and just like learn how they're dying. Also like their anatomy on the inside, because who gets to cope in the sea turtle every day? It's pretty cool. And then mentorship programs. So there is a program called R.I.S.E, and we got to mentor the kids on doing their own research projects, coming up with their own scientific questions and how to answer them and explore data. And that was really cool because I remember when I was doing that and starting out, and it can be daunting at first. And so we got to help them and walk them through that. We also do a lot of events, so fairs and open houses. I got to sit outside a JAWS screening and teach people going in about sharks, but also their prey that we deal with, so marine mammals and sea turtles. And answer questions, raise awareness and that, and then we do first responder training, so for people in the Coast Guard or like public, like EMS or people working on the water, as well as like local citizens, if you come across a dead, stranded marine mammal, sea turtle, alive, dead, or like a whale floating out at sea, like who to call, what to do, what not to do, and just increasing awareness on that front. And then what I highlighted here was beach cleanups. I was able to participate in many beach cleanups, and they're actually what drove my research project, because when we do beach cleanups, we go out with a group, we collect the trash itself, but then we report the data, whether that be the weight of the data, the number of items per category that we're picking up, like bottle caps, straws, how many of them, where, when. So a lot of my work this summer was putting that information into our database, and that database is full of a lot of information, and I wanted to explore what it says. So my project was marine debris analysis of water beaches, and the questions I was asking were, has the amount of debris collected by AMCs changed over time? How has legislation impacted the amount of specific marine debris littering on beaches? And lastly, does the most common type of debris vary by beach? And important when I go into my graphs and everything, the years that I chose to explore were 2017 to 2023. I excluded 2020 for obvious reasons. People weren't going out, weren't doing beach cleanups, so there is no data. And then I chose to explore the beaches that AMCs consistently goes back to and are performing beach cleanups at. So that includes 3 Nassau beaches, Lido, Point Lookout, and Jones, and 5 Suffolk beaches, Halleck, Cupsaw, West Meadow, Sunken Meadow, and Cedar Beach. And to answer the first question, yes, our data shows that of the total amount that we are collecting has changed. So in 2017, AMC collected 220 pounds and then jumped to 2021 and they're collecting 3,523 pounds. So we've definitely ramped up the amount that we're collecting, and that also can be a reflection of effort. So prior to 2020, like those early years are when AMCs was starting out. So maybe they weren't doing as many events, maybe they didn't have as many volunteers, they didn't have their name out there. But now we're collecting way more, and that's a reflection of we're doing more, we have more people involved, and we're collecting more trash. And then for the next part, the second question, the legislation that we do have in place right now on the state level is the 2020 Bag Waste Reduction Act. So that's targeting plastic bags. It's prohibits the use of plastic carryout bags for like stores and food distributors. The styrofoam ban is a 2022 polystyrene foam ban. That's also state level. For Suffolk County in particular, we have our own local laws, which local law 2019 targets straws and stirrers. They're only to be made available upon request to limit the amount of straws entering the marine environment. And then they also in 2019 put a ban on intentional balloon releases. So to start off, plastic bags. So in 2017, we collected 243 plastic bags between all of those beaches I listed before. And then after 2020, we're collecting nearly 3,000 in 2021, but that number has stayed over 2,000 after that as well. So that red line is going to show like where the legislation was placed, and so the before and after. So we are collecting a lot still of plastic bags off the beaches. Because of the ramp up of our efforts as an organization, I don't think that we can from this say that after legislation was placed now, like it's worse than ever, but we can see that we are still collecting like 3,000 bags a year and 2,500 like last year. And I think there's already so many this year when I was exploring the data. So not to say that legislation has made things worse, but I think we are still collecting a lot of bags. For styrofoam, that's gonna be the red dash line is where the polystyrene foam ban went into place. In 2021, we collected 1,287 styrofoam fragments. Then it dropped off in 2022 a little bit, and now in 2023, we're back to like a similar area of numbers like 1,583. So I think it's too soon to tell because this ban went into place in 2022. It's too soon to see the effects of it, so it'll be interesting to see over time how this changes like next year, the year after. But styrofoam is still very prevalent on Long Island beaches even after this ban. Straws and stirrers is a Suffolk County local law, so it was put into place, I think, January 1st, 2020. And as you can see, we're still collecting a lot. That was a huge part of what I was finding over the summer. I would walk the rack lines and find probably without even trying, like I'm just taking photos of the event and I'm picking up things as I see, and I was able to pick up probably like 10 to 20 straws per beach cleanup within an hour. And it's a problem, and we're still finding so many of them, like in the thousands every year. And I think that the exceptions and the way that the laws are written as of now aren't really doing the best that they could for minimizing that entry into our marine environment. For balloons in Suffolk, Suffolk is the only one that has this legislation. Nassau doesn't for intentional balloon releases. So in 2021, we collected 479 balloons, 2023, 351. And then prior to 2020, when that ban went into effect, we were collecting like 200, 100. So once again, I don't think it's fair enough to say, oh, after the ban went into place, we're seeing more and more. It is a reflection of the amount of effort that has increased for our organization, but still like 300 plus balloons that we're picking up every year, and like going out and finding 5 balloons at the beach every time you do a cleanup is still too many balloons to be finding. So these are some bills in progress. They're currently in committee in both the Senate and Assembly. So the Senate bill S2262 is going to create a statewide law against intentional balloon releases, like the one that's in Suffolk. And then the Assembly bill A90A is going to restrict plastic straws and stirrers the same way that the one in Suffolk does now, but just for the state level. So keep your eyes out for that. They are in the initial stages, but I do think it's key and very important of, you know, expanding these laws beyond just the Suffolk level. And then the leaderboard to answer question number 3 about comparing the different beaches and if the type of debris varies. Out of my paper and like all my investigation and analysis, for balloons, it's a tie between Halleck State Park and Lido Beach. Plastic bags, Lido Beach wins. Food wrappers, Lido Beach wins. Fishing line, Halleck wins. Styrofoam, Lido Beach wins. And then fishing nets, Lido Beach wins again. So really, Lido Beach is the overall leader with an honorable mention to Halleck. But effort versus reality. So I don't know if that is something that we can say is like a definite that it does vary between beaches, because we host regular beach cleanups at these 2 beaches, so they kind of bias and sway the data that we currently have. It influences the graphs because they're taking up a majority, but that's could also just because we're going there more and picking up more at those beaches compared to the others. And then, like I said, and I alluded to, after 2020, there appears to be a stark increase in collections, but that's not necessarily a change in the debris on the beaches, but rather maybe just effort. And then areas to improve my study and future studies, create graphs for effort, just to better understand like how that impacts the data that we have, but also to like see like how people are interacting with conservation and, you know, the interest that we're gaining from the community. Things like that that could really like help like forward legislation and, you know, expand conservation measures. Expand cleanups, so we're hosting cleanups at similar beaches all the time, just because we know people there want to do it, and we have our own partners there. But we could do cleanups everywhere and do them consistently, so that we're not cleaning up the same beaches, but we're expanding to all different beaches. Continuing to collect data after these legislation goes forward, like, for example, the styrofoam ban started in 2022, so if we continue to track the data, maybe we will see a change, maybe we'll be able to see how that's impacting the amount of debris on our beaches. And then also, I didn't put it up here, but also, you know, sharing data between different organizations. We're not the only ones doing beach cleanups. There are so many organizations on land doing them. So to share our data with them, maybe if everyone had one communal database and we could see patterns a little bit more and be able to analyze that a little more consistently. And then I'm sorry you couldn't be there in person, so I won't be able to answer questions, but if you do have questions about like my internship or my data, anything interesting that I found, this is my email address, just shoot me an email and I'll get back to you. Yeah, but thank you, everyone. Thank you to AMCs. Good luck to everyone entering the program, and I wish you the very best.
Our next speaker is joining us by Zoom. And it is Anna Penavic.
Yes, and she is gonna share her screen.
She's gonna share her screen.
Yeah, Anna, go ahead, whenever you're
Hi,
ready.
Anna.
Hey guys, how's it going? Can you hear me okay?
Yeah, we hear you perfect.
Yeah.
Okay, great. I am in a public setting, so hopefully you guys can hear me okay throughout the duration. So sorry about that. I got stuck in Munich, like my flight got messed up, so I'm here for the day. So gonna try my
And
best.
Anna's already started a job, which is a big part of why she's not here today.
Yes, but I did get vacation from my job, so that's why I'm now flying. Okay, so hello, my name is Anna Penavic, and my presentation today will be about a study on giant clams, Tridacna species, abundance, and age structure in Koh Phangan, Thailand. So my internship supervisor was Victoria Fahey at R.I.S.E, and Dr. David J. Taylor was my advisor at SoMAS. Just today, I wanted to introduce giant clams to you guys, and I want to go over my research objectives, as well as introduce R.I.S.E to you guys, and introduce the region where I was in Thailand called Koh Phangan. I'll also be going over the methods of my data collection and going over the data collection as well as the results, and then I will discuss everything that I found, as well as future research that R.I.S.E is considering doing, and just other things I found from my data collection. And then I will conclude my presentation. So just to introduce giant clams, they are the largest bivalves and found in the world, and they are vital to coral reef ecosystems. They're found specifically in the South Pacific and Indian Oceans, with 3 species specifically found in the Gulf of Thailand, which are Tridacna gigas, Tridacna maxima, Tridacna crocea, Tridacna squamosa, and Tridacna maximum. And they have a symbiotic relationship with the zooxanthellae that live within their mantle, similar to sclerotinian corals. So I don't know if you can see in the photo, but they're very vibrant. This is a more white and brown colored one, but other slides you'll see how vibrant they are, and they're very beautiful. So it was really amazing to be able to dive and do this project on them. So you can see also, so all the photos are from my research, and you can see the transect line in the corner here. So I'm going to be going over my data collection later on, but just to point out the photo to you guys. And so the motivation for this was to study more about giant clams, to understand and protect this endangered species, because they are listed in CITES under Appendix 2, and specifically by the Thai government, by their Wild Animal Reservation and Protection Act BE 2535, which was enacted in 2014. And specifically, giant clam populations are decreasing worldwide, and that could be due to overharvesting. Since they are so beautiful, they are collected for aquariums. Their mantles also, they're very large. They can grow up to 1.2 meters in size, and there have been weights reported up to 237 kilograms. And so their shells are just very beautiful. And so they're also are harvested for those reasons, just for aesthetic purposes. And their meat is eaten in Japan, so sometimes their meat is used for that as well. And so climate change and rising sea temperatures is affecting them, much like hard corals throughout the world. With the zooxanthellae, once there's past a certain temperature threshold, the zooxanthellae do get expelled, and so they do bleach, and as well as human pollution and any runoff coming from shorelines, so they do get affected by anthropogenic pollution. So my research objectives, I had 2 questions I wanted to answer by collecting data about giant clams. And question number 1 was, does the abundance of giant clams differ between coral reef habitats, so reef crest or reef flat? And question 2 is, what is the size in relation to age structure of giant clam populations in the reef flat versus the reef crest? And so I just wanted to talk first about C and where my internship was. So they are the Center for Oceanic Research and Education in Southeast Asia, and they're a nonprofit organization that was founded in 2013 by a German guy named Eike, who really wanted to look at preservation of marine biodiversity. He wanted to actually first do it in Mexico, but there's such a big organizational presence there for that already that when he went to Thailand to do his dive masters and dive instructors training, he found that there was nothing there, and so he started it there. So C is really vital to the community on Koh Phangan for the work they do and the education that they do and the outreach, just to teach the community about how to take care of the area that they live in. So specifically when I was there, there is a lot of Burmese refugees, since Myanmar is going through civil war at the moment. So we were tasked with creating a video to help introduce just like the sea world to the children of these refugees, for them to really just know what's out there. And that's in partner with a local organization that teaches them how to swim, because a lot of people actually don't know how to swim, right? So that's an important thing, a first step, I think, to understanding and engaging with the water world. My study locations, so Koh Phangan is located in Thailand, which is in the Samui archipelago, and so Koh Samui is the biggest island found in Koh Phangan. It's known as the Ibiza of Thailand, and Koh Phangan is a small island located above that. There were 7 locations that we collected data at. So specifically when I was there, I just did 3 months worth of research collection. But my internship director was kind enough to give me the data since they've started collecting data about giant clams. So I do have all the data from March 2022 to June 2024. And so Had Yao, Had Salad, May Had, Koma, Ao Tong Lang, Ao Chalaklam, and Had Kom were the 7 locations that we did data collection at. I specifically did data collection at Had Salad, May Had, and Ao Tong Lang. And I did dive at Koma, but did not do any data collection there. And so we are based in Chalaklam, the town that's right below Chalaklam Bay East, which is Ao Chalaklam. Fun fact, Had Salad actually means Pirates Cove, because that's where all the pirates would dock to sell their wares here. And it's a really beautiful area. And yeah, so methods and data collection. So we conducted scuba surveys every single day, 1 per day. Every morning we would have our daily meeting, and Victoria, as you can see, actually her fin labeled Vicky, would tell us where we were going. So let's say we went to Mayhad, she would tell us if we're going to the reef flat or the reef crest. And so she would split up teams by the data we were doing. And we always had 3 transect lines. And so that were each 50 meters long at each site. So a total of 150 meters length with our transect data. And like I said previously, I collected data from March 2024 to May 2024, but I have the data from March 2022 to June 2024. And so reef flat was 2 to 3 meters typically at depth versus the reef crest, which is 5 to 6 meters, which is really interesting to note because typically the reef crest is the area that crests, but Koh Phangan, for some reason, the reef crest just declines in depth and does not peak up. So that was just really interesting to learn to dive at and like know that. So I just wanted to give a survey example of when we collected data. So typically I would have either a partner helping me collect the data or I would do it myself. And whatever, the belt line would be 2.5 meters on each side, so a total of 5 meter a belt from the transect. And whenever we would see a giant clam, we would measure it and just write it down on our slate. And when we came back to the property, we would go to the lab and then just input it into the giant Excel motherboard sheet that we had. And so we always recorded the location, the visibility, the depth, and what zone we were in. And we would differentiate between what transect we found the data at. And so for the results, I started off by looking at the total surveys conducted and the total amount of clams that we found. And so it was a great number, and it was 3,505 clams were surveyed in total for the 3 years. And 3 years, yes, 3 years. And as you can see, Hadsalad only had one survey done. And so when I did this, it was easier for me actually to sparse the data by just doing total survey dates, because I was really struggling with differentiating it between doing survey date with transect number. And so I did the total measurement as 150 meters for the rest of my data as well. And so Mayhad was one of our more popular study sites, and we found a total of 774 clams there over a course of 23 surveys. And Hadsalad, the crest, the flat had only one survey done, but there was still a good amount of corals found there. Giant clams found there. And so for giant clam abundance, Koma had the least. So the crest overall, the reef crest had the lowest overall population density versus the reef flat. And so Koma had the lowest density at 0.0155 individuals per meter squared. And so like I had said, when I was doing the data sparsing, it was easier for me to do this over a course of 750 square meters instead of doing it individual by transect line. And so Mayhad 4 had the highest population density at 0.2775 individuals per meter squared. And as you can see, Hadcom, Hadyao, and Koma at the reef flat did not have any data collected because we just weren't diving at those sites. And so we typically, like I had said, when I was diving, it was Aotonglan, Chalakambe, Hadsalad, and Mayhad 4. Those were like our primary sites. And so even for Hadsalad, the flat, there was only one survey conducted. So that's why there's no standard deviation for the reef flat and Hadyao, just because it was only one survey collected. And so for the age structure, so when I say age structure, when we were measuring the size, that correlates to age structure. And so as you can see, there's a decline in sizes for both the reef flat and the reef crest from 2022 to 2024. And the regression line, the linear regression found that for the reef crest, it declined by 1.55 units per year. And for the reef flat, 2.96. Oh my God, I can't speak, 2.95 units decline per year. And so for the p-values, there's significant decreasing trend because both were less than 0.05. And can you guys hear me okay? Sorry, and I know I'm talking and I can't see anyone, so I'm just talking to the wind, but
yes, we hear you.
okay, I'm glad. I should have asked that earlier, but and so for the size range, I just thought it would be interesting to compare the ranges and the values and everything. And so 2023 found an interesting year. Saw interesting numbers because the reef flats had such a high minimum number and such a low maximum number, even though all the other years were typically similar values, and then for the minimum values, 2 centimeters. And like, they're so tiny, it's so funny. Like when you see them, you're like, oh my God, like it's amazing. Sometimes you can really miss it. So I think a lot of the data for the limitations were actually in the amount of interns that were collecting the data and like being able to see the clams and actually differences in measuring, just because the clams curve. And so sometimes it was difficult because you're in the water, if you're not really great at buoyancy control, like how can you really measure it? And if you like crest along the giant clam, sometimes your data values could be larger and smaller. So I feel like typically when I was comparing my data to other people's, I always had lower values because I think I was really measuring more so over the giant clam than following the clam's shape. But yeah, I just wanted to point out the size ranges. So yes, I'm at 10.
You're at 12 minutes, so just try and.
Okay, sorry. Okay, well, so I wanted to say that there is significant negative trends in clam size because the p-value is less than 0.05 in both habitats. And there's a higher clam abundance in the reef flats, possibly due to better environmental conditions. And that the decline in clam size does indicate potential stress factors such as an anthropogenic stresses and environmental stresses as well, such as increasing temperatures. So future research could be looking at, do certain Tridacna species thrive more in Koh Phangan? Because that's one thing that we didn't do, is we didn't differentiate what species we were seeing of the 3 species that are found in the Gulf of Thailand. And so other questions were, do specific species fare better within increasing temperatures? So we were actually talking about potentially measuring the bleaching found in the giant clams. And that's something I think Porites is considering doing in the future. So doing a giant clam bleaching study, and other specific zoos on belly that live within giant clams that fare better within increasing temperatures as well. And so I just wanted to conclude, and this is a video of my friend and I diving. It's really loud, sorry. Oh, well, I just wanted to say, oh my God, I can't pause it. I can't pause it. All right, it's gonna have to play out, but oh, I can. Okay, great. So this study provides valuable insights into giant clam populations in Koh Phangan, and it emphasizes further need for conservation efforts to protect these vital reef species, because of their beauty, but also because of their filtration systems that they utilize in the region. So, and tourism purposes as well. So thank you so much, guys. And you can continue watching my friend and I making fools of ourselves. But yeah, thanks so much. Those are my references.
Thank you very much, and congratulations on your position.
Thank you so much.
What's the job that you'll be doing?
So I have been working with them throughout the summer. I'm in New Hampshire. I'm working with a dive company, and we're removing invasive species called milfoil typically. And so we just dive in to the sites that the DEC sends us to, and we just remove the invasive species. So it's been great. I've been diving every day, so it's been really nice. Thank you, David. I appreciate it.
This is the last virtual presentation. All the others will be in person.
Yeah.
We kind of put them together in the middle.
Claire is still at her internship in Curacao, so I guess that's a good reason to not be here.
It's a nonprofit organization that focuses on coral reef restoration. It's very tiny. There's only 2 full-time staff, and they have internships for interns who kind of rotate and help them out. My project was based on kind of the previous intern's project, which was with Porites species. And while they were documenting those Porites, they saw a lot of really weird morphologies that they could not figure out what they were or why they were doing what they were doing. So my project was all about looking into that. And okay, so these on the picture underneath the question marks is just one example of the really weird morphs we are seeing. If it's anything, it's most likely Porites, but the big difference here is that Porites are branching coral, and the weird morphs we're seeing are more encrusting and mounding. A lot of people have also dismissed it as Porites, but if we look at these 2 pictures, they're just obviously not the same thing. So that's not the right explanation either. So my project consisted of documenting all Porites species except for Porites in the shallow waters along breakwaters on the leeward side of the island, to try and figure out why these morphs exist and what caused them to exist. We had 3 hypotheses to why we're seeing these weird morphs. The first one was parrotfish predation is causing local adaptation. So these morphs are a result of the parrotfish predating on the coral, which is causing them to change the shape to hopefully avoid more predation. Our next hypothesis was depth and wave action affect morph. We saw these strange morphs in really shallow waters where the wave action was really high, which made these transect surveys really, really hard. And these conditions are not ideal for branching coral, so this does make sense. And the last hypothesis, which is our favorite after all is said and done, is that these abnormal morphs are actually a new species that we haven't heard of before. So this project is actually a prequel to the big project that R.I.S.E wants to do, which is to look at the genetic and molecular makeup of these morphs and compare them to Porites species that are well studied around the island. But to do that, that requires funding that my internship organization doesn't currently have. So to prove significant chemical functionality, we have to first prove significant ecological functionality. To prove that there's something happening that requires more funding and more research that our small organization isn't capable of. So to prove ecological functionality, we conducted 9 transect surveys across 5 different locations. And for each colony along the transect, we recorded depth, morphology, predation, upward growth, diameter, its donation on the substrate, if it's associated with fire coral, and if it's in a cryptic location. And our next step is to look at reciprocal transplants, which we're kind of still trying to figure out, to look at response to local adaptation and predation. And then once all that is said and done, we can use those findings to request grant money for genetic and molecular testing, and then see what the results say. So morphology, predation, and depth were the three biggest data points we wanted to collect. They're the most important to us. So morphology was grouped into 6 categories. First, we had encrusting juvenile, which is encrusting but just smaller than 2 centimeters. And then we had encrusting, which is just bigger than 2 centimeters. Then we had stubby non-furcate, which looks like that. I don't even know how to describe it. Our next 3 is bludgeon, which looks like you can attach it to the end of a stick and whack somebody with it. We have long non-furcate, which has arms but no branches on the arms, and then long furcate, which has arms and branches on the arms. These long non-furcate and long furcate are more of the normal morphs that we're looking at. And then the bludgeon, stubby non-furcate, and encrusting are the strange morphs. Predation was also categorized into 6 groups. We had non-existing, so no predation on the coral. And then we had fresh tissue and skeleton removed from the coral. And then we had just the tissue removed, but the skeleton was still there. And then we had tissue healed, so the polyps have returned, but there's no color, which means the zooxanthellae algae hasn't returned. And then we have old tissue color returning, where some of the algae is back, but it's still getting there. And then lastly, we had algae overgrowth, which means instead of the tissue returning, algae grew in that dead spot, so that spot of coral is just no longer exists. So we, in about a month and a half, we looked at 291 different coral colonies, and I will go over the significant results from the test that we did. Sorry if it looks like I'm sweating. It's like 95 degrees here every single day, so I'm really hot. Anyway, when we're looking at morph versus depth, there's clear overlaps between all the different depths at the morphs, but there was significant differences between encrusting juvenile with bludgeon, long non-furcate, and long furcate. And then we also had a difference between encrusting and long non-furcate. We can see from this graph that the branching corals tend to grow deeper than the encrusting corals. So that does support the hypothesis that depth does impact morph, but that's not fully supported because of the amount of overlap we're seeing. And it doesn't really make sense for 2 coral colonies of the same exact species to grow at the same depth, and in some cases directly right next to each other, to have different morphs, unless of course they're not the same species at all. So there's something to this, but there's also more to the story than just depth. Our first leading theory was that predation was causing these morphs. We kind of decided after looking at this data that that's not the case. There was some significant differences between encrusting juvenile with encrusting and bludgeon, and that could be explained by the encrusting juvenile's small size and the increased likelihood that it was settled in a cryptic location. But if predation was the cause of the morph, then we would see a lot more predation on the branching corals and less on the encrusting corals, because the encrusting is the new morph. But that was not the case at all. So it is unlikely that predation is the cause of morphs. And for our predation versus location test, we saw the most significant differences at one site, Parasasa Reef, where we did 4 surveys. Because even though these survey sites were really close together, the majority predation type was different at each site. So that does show that predation or location has some effect on predation. This could be a result of just the environment, like nutrient loading, nitrogen, other factors, but that's unlikely, especially in this case, because the environment was so close together to the other environments. So the other explanation could be parrotfish behavior. Parrotfish are very selective in the coral colonies that they predate on, and male terminal phage parrotfish are territorial, so they let a bunch of females into their territory, but not other males, so that can also affect predation as well. So lastly, I'm going to talk about how all this research connects to marine conservation. So if this is in fact a new species, which would be so cool to be part of finding, that increases the biodiversity, which means the reef is healthy and it's doing good. However, if it's not a new species, coral morphology is an indicator of habitat complexity, rare species occurrence, and species richness of a reef. And all those things impact the protection status of a reef, because the more of those things that a reef has, the more likely it is that they're going to be protective. So this research can help some reefs qualify for protection that they don't currently have. So that can possibly impact legislation around the island and protect these corals and the reefs that they're living on, which can indirectly affect coastal development, which there is a lot of happening on the island. Most of the beaches are man-made. A lot of the breakwaters are man-made. Currently, the biggest underwater restaurant in the Caribbean is under construction, a kilometer away from where I work. So while coastal development and legislation was not the goal of this research project, it is a happy side effect if our findings show that more protection is warranted. But our main goal was just to look at these corals and try to understand what they were, why they were doing, and why they were where they were. So we still have more work to do. This is just part one that we just finished. And our next step is to look at reciprocal transplants and to see more about predation and local adaptation, because that was our first big theory and we're not ready to give on it yet. And we can't really go into our next big theory, which is new species, without the funding and answers from the reciprocal transplant. That's it. Let me know if you have any questions, or if my Zoom is bad, you can email me at this email here as well.
Great. We do have some time for questions. Anyone have questions for class? Juliana Dovi is next, and her capstone advisor is Ann McElroy. I don't see Ann.
No, she wasn't able to come today.
Okay, so she wasn't able to come. And she's going to be talking about the importance of an in-person scientific workshop.
All right. So my presentation is called The Advantages of Discussing Difficult Environmental Concerns Through In-Person Workshops Aimed at Scientific Collaboration and Education. So a mouthful. My mentors are Dr. Ann McElroy, who can't be here today, and Dr. John Cortez. He's actually based in Columbia, so definitely can't be here today.
Oh, hang on, my fault. You should be able to do it.
Okay, so I'll be discussing blueprints for resilience, but first, what is that? This was a workshop hosted at Stony Brook University from June 10th to 13th. The 4 days, 2 of them were actual workshop days where we sat in this room. Dr. Pikitch was actually part of this group, so she can even attest to that. It was funded by an NSF grant, National Science Foundation, called Defining the Role of Steroids in Temperature and Coral Reproduction. The PIs on this are Dr. Kleinhaus, Dr. McElroy, and Dr. Klein in Israel. And the point of this grant was actually to encourage collaborative work between US and Israeli scientists. And the broader aspect of this workshop was to go towards broader impacts, which is a requirement for NSF grants that we have some kind of broader impact requirement for what that grant money is going towards. So basically this means it has to affect more people than just the actual scientists working on the research question for the grant fund. It has to go towards helping broaden the research community and maybe even the public. There were 17 invited guests representing 6 countries and all these organizations mentioned, and there were 5 participating organizers, including myself and 1 other MCP student. During this workshop, we pretty much split into 3 groups, including the R.I.S.E, the Caribbean, and a general global overview group, which I'll discuss in a moment. So of this, 6 attendees were junior colleagues, and actually a big goal of this workshop were to invite some of these younger students. These were typically people that were pursuing their PhDs, so in their PhD years, and everyone again was encouraged to bring somebody along with them. This was to get them into that broader conversation of science. Maybe they were never a part of these larger workshops, and we really want them to especially help out in the later goals of the workshop I'll mention later, but it was important for us to get these younger scientists in the conversation. 11 senior scientists presented on their work. This was super important just for everybody to get to know each other during these sessions. And of course, a Q&A session followed that. There are 4 main objectives of the workshop, which I can't review for sake of time, but for example, one of the objectives was pretty much to outline every problem that coral reefs are facing. And each of these objectives had activities to guide this effective discussion. So what was my role in all of this? I actually started workshop preparation in the spring. So I've been working on this for a few months before the actual workshop took place. I helped pretty much with a task every week we meet. A task would occur and I would have to submit this task by the next week. So one of these tasks would be creating a reference library of all of the 4 objectives that we had. Each of these objectives needed something like 10 to 15 background papers that we can pretty much back up that research and hopefully again use this in our more official publications in the end, resulting from the workshop. I also drafted a primer paper which was sent out to all participants. This basically just outlined what we're actually going to be talking about and use some of the reference paper libraries in this as well. I collected attendee information, so discussing one-on-one with every attendee, just a little bit more information so we can create a roster so everyone knew who everyone was beforehand. I, of course, attended and helped logistics of all workshop activities and used detailed notes during all the workshop events. So my specific project for the MCP program in this whole workshop here is what makes in-person workshops valuable. So I really took a specific focus on the effectiveness of workshops. Again, for the sake of time, I don't have time to discuss a big chunk of what my paper was, which was reviewing previous workshops and discussing what factors might make them successful. This is focusing on their workshop goals, how they're accomplished, some outcomes of the workshops, and then, if available, participant feedback. For this presentation, I will go over the second half of my paper, which is reviewing blueprints for resilience. In my paper, I, of course, compared them to those previous workshops to see what we did different and similar. And of course, we had a review of an exit survey. So what was the significance of our events? What made our workshop in R.I.S.E successful? So first, we had participants with regional expertise. Like I said, we split up into 3 groups, the Red Sea, the Caribbean, and a global group. And from here, it was really important that before the workshop started, we had participants coming from all these regions. We wanted these regions focused on, so at the end of it, we can put out an outline plan of what we're going to do to help that specific area. And these scientists were able to use their regional expertise within these groups. We had activities during the workshop that was aimed at group collaboration within these regional groups. Some of these activities include systems mapping. Just a sentence about this. This is basically creating feedback loops that we can actually write down and kind of understand how one effect on coral affects the next. So we actually used a really neat application called Miro. It's an online collaborative, basically whiteboard. Everyone could work on it at the same time, but in your group, you can discuss what you're writing. And this was a really effective activity during the workshop. We did a force field analysis. This is basically us taking all of the different problems we found coral reefs were facing, understanding which stakeholder groups were going to be the most effective in helping with these different issues, and then the effect that we had on those stakeholders as a group. And then we created graphs from here, which we can actually see tangible evidence of which stakeholder groups we should target the most with our outcomes. And then we also had World Cafe sessions. The point of this was to pretty much encourage collaboration between the different regional groups. That was another big aspect that we wanted. Not only should we have these regional plans, but groups should intermingle and pretty much discuss and give their ideas on what that group thought for better and worse. And we also had social receptions, dinners, and a field trip to Governor's Island. I think this is a super important aspect. This really helps create those informal working relationships that will hopefully benefit in the future. And then overall for the workshop, we really hope to have a white paper and a few peer-reviewed papers coming out of this, hopefully led by key participants of the workshop. We hope to have created collaboration between attendees. We really wanted this to be a really unique knowledge sharing experience. And it has to be an opportunity for, again, those younger graduate students to get in the high-level discussion. So I discussed some of our survey feedback here. In this chart here, you see 4 questions asked about if participants thought that education was increased, collaboration, workshop materials were useful, and if we thought objectives were achieved. And I think the biggest takeaway here is that no negative responses were pretty much submitted for that. And we also see 90% of the responders agreeing that the workshop benefited their coral conservation knowledge, kind of indicating to us that at least some of our participants thought it did act as a knowledge exchange opportunity. I should have mentioned before, this is a 15-question, super simple survey. Most of the questions were multiple choice, just so we can hopefully get the highest amount of replies back. And of 17 invited guests, we had 10 respondents. So that's a little over 50% of what we have here. Our next graph here is pretty much showing what attendees thought was valuable versus what could be improved. And we see pretty much interaction with participants was the most positive aspect of the workshop. We also see presentation and discussion sessions having equal amounts of praise and need for improvement. So this is kind of a confusing result. Is this something that could be a flaw in the survey? Should we follow up with a separate survey? Or is this just different opinions of different participants? And we see that reflected in the results. On this graph here, pretty much discusses organization and timing of workshop components. We see 90% of participants responding that they were either very satisfied or dissatisfied by the timing of workshop components. But when we compare this to the pre-response section, this is a little interesting. I don't have time to talk about all the pre-response, but I will highlight some of the key findings. 4 responses indicated the need for more time. This could be typically, many of them said 2 to 3 extra days. But when comparing it to our other graph there, it said 90% were satisfied by it. So these are kind of conflicting results. The need for more participants was apparent. And something I think that was completely evaded by all workshop organizers is the need to declare English as the official workshop language. I think many of the organizers just thought it was obvious. A lot of the presentations and all group discussion was held in English, but obviously this survey pointed out that somebody in the room did not feel that way. So definitely something to look forward to going forward if another survey was to be completed or another similar workshop. The need for more World Cafe sessions, so intermingling between those regional groups, was definitely a point brought out, and an outline for next steps going forward. So not everybody that left the room fully understood what were the next steps on a person-by-person basis. We all knew that we wanted a white paper, we wanted some peer review papers, but not everyone was sure how they're going to be playing in that role. But overall, we see with our survey, of the 10 people that responded, 100% of them said they would attend a similar workshop in the future. So we think that's a pretty good result. And what does all of this tell us? Overall, the survey really helped understand some participant view on success. Discussion was an obvious highlight for blueprints for resilience. We do need more attendees and longer time, but for this, there are definitely limitations. With our grant fund, we don't know how much money that can go towards, and really how much time can people get away from their regular life to come to our workshop. The need for increased collaboration after the workshop to make sure all participants are on the same level, knowing what's going on next. We do have a Zoom meeting already set in motion for September, and regular follow-up meetings, of course, after that for publication creation. But we really want to make sure that everybody left the room knowing what their steps are next. Overall, my opinion on the workshop, I think it was a super great opportunity for someone so young like myself, who's never really been involved in this high-level discussion, to get involved. I think watching all these interdisciplinary scientists from pretty much all over the world work towards a greater goal was a really, really interesting aspect on this workshop in general. I have a lot of thank yous to do that. So thanks to both of my mentors for really helping me through the whole process. Thank you to representatives from Duke University, really helped facilitate the workshop and help me create the survey to get the most out of our answers. Thanks to Dr. Crane Kleinhaus for workshop organization and grant work. And of course, thank you to the National Science Foundation for our grant and for funding my research paper. Thank you.
That's great. Okay, well, we do have some time for questions.
Yeah.
There's a question.
I'm just curious if you have a sense for how many people could have attended a Zoom workshop but weren't able to attend in person.
Honestly, the whole point of this workshop was to have an in-person discussion. So I don't think that was even on the table to have a Zoom discussion. They really focused on getting people in there in person. So
I'm just
yeah.
wondering if there were people that declined, like, you know.
There definitely were, because everyone, and there were even people that we wanted to invite from Australia, say, and tickets are just so expensive to fly them over, and we don't have enough money for that. So that could be an option, but a big part of the goal was to get people in person to grow those working relationships. Can't really do that as much over the phone.
Question?
Yeah.
When did you go to Governor's Islands?
It was the last day of the workshop, so June 13th. We had the New York Climate Exchange explained to us. We got to learn about Billy and Oyster Project. It was a really great time for people that have never been in the area.
Question? Okay. Thank you very much.
Hi, everybody. For my project this summer, I decided to do a literature search on the effects of coal ash on water quality and public health. My mentors were Dr. Collins and Dr. Chris Goldberg. So I started looking at historical records at first. So I was able to find records as far back as 1949, courtesy of the U.S. Energy Information Administration. Some highlights of what I found: coal made up 16.2% of total energy consumption last year, compared to 48.5% in 2007, which is the peak of coal usage in the US. It's important to note also that during 2020, after COVID, coal usage dropped substantially, and it actually is increasing from last year. So it's on the rise a little bit, but not quite so much. It's been replaced mostly by natural gases and renewable energy sources. The US consumed 594 million tons in 2022. Peak production in 2001 of 1.13 billion tons. So there's quite a lot being used over the years. Just an overview of the process. So during the mining process, coal deposits must be separated from surrounding rock and soil. Water and gravity are used to separate the ore deposits from the surrounding soil. In the picture below, that's a coal washing plant. They're separated based on density, so they're able to absorb the biomatter more easily, which is the coal, which is what they want. So the more that they would rinse it and wash it, the purer the coal is. There's less other sources in it that they can't use as fuel. And the more pure the coal is, the higher price they can get for it, which is what they want. So it's in their best interest to clean it as thoroughly as possible. So just a highlight of the burning process. So for about every 7 to 10 tons of coal burned, about 1 ton of coal ash is produced. The rest of it is released as CO2, which we all know. Coal ash is made of 3 general constituents, being fly ash, bottom ash, and boiler slag. The most important one is the fly ash, because it's the largest volume of the waste that's produced, and it's the most useful in terms of being able to be recycled as well. But the lower the purity of the coal that's used, the more coal ash that is produced. So this is known as coal combustion residuals that the coal ash that they're producing. In terms of storing the coal ash, it can be placed in landfills, or in a solid form, or it can be placed in coal ash ponds by adding water to it to make it a liquid. The benefits of being a liquid is it doesn't get aerated into the air as much. So it's less of an issue for people to breathe in. So that's one of the positives of that. But the coal ash ponds are left uncovered, and most of them are unlined, so it's easily leached into the ground beneath it. So across the US, it's a little bit debatable, but there are at least 400 sites within the contiguous US and Puerto Rico. There's one in Puerto Rico on the southern shore. Most of the ponds are very large, being thousands of acres wide. And I'll talk more about recycling options later. So this is just a picture of the distribution of the coal ash ponds and landfills. So the black ones are the unlined units. The yellow are the liner status not being disclosed, and the green are lined units. And you can see most of them are not green. So some of the components of coal ash, they're combined with waste that's produced during the mining process, and they contain a laundry list of heavy metals and other harmful chemicals that you would not want to interact with. For my project, I'll be talking mostly about selenium. That's where it seems to be a lot of the aquatic issues surfacing to do with selenium. So some interesting things I found in terms of public health. So coal power plants are built next to fresh water sources to facilitate easier disposal, which is convenient for the coal mining corporations, but not so good for us, because that's where most people live, near a water source. Because they're left open to the elements, engineering failure is common. Storms and other environmental effects can lead to flooding, and they can cause the coal ash ponds to leak into the surrounding area. And I'll talk more about a case later. People who live in the US, people who live within 0.5 miles of coal power plants are more likely to experience negative respiratory health symptoms, especially if they spend more time outdoors. One study I found by Moram et al. From 2019 says that while Black and African Americans make only 13% of the national population, 68% of people living within 30 miles of coal plants were Black or African American. So I just thought that was interesting to highlight and should be talked more about. So my first case study is the Kingston Fossil Plant in Kingston, Tennessee. This power plant failure occurred in 2008. It involves a 6-story dam that was created with waste from the mining process. And the dam completely collapsed, leading to the whole pond leaking into the surrounding town. So 1.1 billion gallons of coal ash leaked into the surrounding area and into the waterways. 15 homes were engulfed in the black slurry. 900 workers were dispatched to clean up the spill over 5 years. There were no deaths or injuries from the initial spill, but they had found that many workers over the course of 10 years after exposure experienced issues, including brain, blood, and lung cancer from overexposure during the cleanup. After 10 years, more than 30 of the workers had died, and 250 suffered from chronic illnesses. And they're also currently undergoing a lawsuit because they were not warned of the dangers prior to the cleanup. And the company responsible, the Kingston fossil plant, finally admitted that there was a problem, and they're in the process of a dispute, but there's been no payout at this time yet. So the picture to the left is before the accident. The picture in the middle is before the cleanup. And on the right from 2014, you can see that the entire area is just completely gray and decimated, basically a lifeless wasteland. So it just is a testament to how much it affected the surrounding environment. So as far as marine conservation is concerned, there are risks to aquatic life. Poisoning can occur directly through exposure or by eating a contaminated organism. The toxin bioaccumulates within the organism and may affect their fecundity, which is their sexual rate. It can also biomagnify within the food chain, and exposure can result in mutations, death, and diseases, which I'll go over more later. So one case study I found on the Caribou National Forest. So many of the Western states have geology with high levels of selenium. So just drilling into the seleniferous rock can result in selenium leaching into surrounding water sources. Coal ash is found to contain high levels of selenium as well. And the level of selenium disclosed by J.R. Simplet Mining Company. During this, so they have a mine in the Caribou National Forest called the Smoky Mountain Mine, and they're mining phosphate there, and they haven't said about how much selenium is contaminating the area. So they conducted their own study to assess how negatively it was affecting the area, and they found it to be not much of an issue. So even low levels of selenium found they lead to mutations in eggs and developing embryos, and they result in high levels of mutation in the Yellowstone cutthroat trout and the brown trout, which I'll show pictures of later. But according to them, the mining was not an issue. And these are some pictures of the mutations. So on the top is the Yellowstone cutthroat trout, and on the bottom is the brown trout. And funnily enough, when they were asked about this problem, they didn't know about this before seeing the pictures. And after seeing the pictures, they said 2 heads are better than 1. Or the more heads, the better, which shows you how much they care about this happening. In another case study in the Bellews Lake, this accident occurred in North Carolina in the 1970s. So coal ash leached into Bellews Lake, poisoning 19 of the 20 fish species found in the 3,800-acre lake. The deformities persisted for decades following, and the selenium was found to be the culprit. I'll show you a picture of that too. So in fish A, there is a sideways spinal deformity, and in fish B, it's more of an upward deformity, and C is the normal fish, and this is called the mosquito fish. So some recycling options. There are a lot of recycling options for coal ash, but nearly all of them still run the risk of it leaching into the surrounding environment. So there is no foolproof method for recycling, but it can be used as a substitute in concrete as cement. So that's one of the bigger ways it can be used. Some remediation. So in 2015, the EPA passed a bill called the Coal Ash Rule, and they had found that coal ash impoundments must have groundwater monitoring systems. Leaking ponds must be closed or retrofitted with liners starting November 2024. So they're going to be closing soon. And the Trump administration did roll back some regulations, so they're in the process of regaining those losses. And complete leaching prevention isn't possible. The EPA discloses themselves that liners only slow the process. So conclusions, reliance on coal is dwindling, but existing waste persists. Recycling methods are not all encompassing. All storage methods have downsides. Exposure to coal is dangerous for people and the surrounding environment. And proper handling of waste is paramount for future well-being. And I'll take any questions anybody has about my topic.
Okay, we do have a few minutes for questions.
I have one. So did you come across any studies that looked at the use of coal ash in cement? Like, does it leach at anything? Or is it as strong as like traditional Portland cement, all that kind of stuff?
Because it's solid, it doesn't leach. It doesn't run the risk of leaching nearly as much, but it's still exposed to the elements.
Right.
I didn't see like a rate of leachate, but I would say that, anything breaks out in nature. So whenever the cement is degraded, some of it gets released into the environment, but I didn't see an exact number.
Other questions? The quiet crowd. Carl?
Yeah.
So what are the implications of the mercury? You didn't talk about mercury, but we're all concerned about mercury in seafood that comes from burning coal.
I did find a lot of things about the bioaccumulation of chemicals within organisms. And so there's a lot of information out there about seabirds eating fish contaminated, and it led to a lot of seabirds dying as a result. I tried to limit mostly to the fish that I found. And a lot of the stories were from, I guess, more historical times, not so much in the recent future, but I looked up some of the chemicals found in that list I gave, and I found selenium to be like widely talked about. And I didn't know much about selenium prior to this research project, so I decided to stick with the selenium. I didn't talk much about mercury, so sorry about that. In the interest of time. It's a lot to cover in 10 minutes.
Yeah, maybe one more.
All right, thank you.
Okay, thanks, Finn. Isabella Imbo is our next speaker, and she was mentored by Mass Botani, Cornell Cooperative Extension. Another organization that has a long history of working with us.
You took the clip with you.
Advising our students. Somebody took the clip?
If you use the clip on your
Oh.
just need the microphone. Lord, thank you.
I was keeping it for
You really wanted to talk some more. There we go.
Okay, so Isabella's talk, from lab to field, investigating substrate preferences in ribbed mussels.
Hello, everyone. Thank you for being here today. My name is Isabella Imbo, and my project title is From Lab to Field. So from lab to field, investigating substrate preferences in ribbed mussels. So my agenda today will begin with an introduction to my project and some background information to help you understand my research objectives, my experimental methods, the results and how we got those results, and discussion and conclusion. So I conducted my internship at Cornell Cooperative Extension, where my focus was on bivalve restoration methods. My work was part of a larger collaboration with scientists from CUNY, Stony Brook, and NYC DEP. We are exploring novel solutions to enhance water quality in urban environments through bivalve biomediation. My particular study involved investigating substrate preferences in ribbed mussels, which play a crucial role in coastal ecosystems. Firstly, ribbed mussels are vital to the health of salt marsh ecosystems. They improve water quality through filtration, capable of filtering bacteria-sized particles from the water column, thus reducing bacterial loads and improving overall water health. Additionally, they stabilize marshlands and contribute significantly to nutrient cycling. Lastly, recent studies have highlighted their role in nitrogen removal, which is particularly important in eutrophic environments like urban estuaries. This filtration is particularly valuable in urban areas impacted by combined sewer overflows, also known as CSOs, where mussels can help control bacterial contamination. Previous studies show that large populations, around 50 million mussels, are needed to significantly impact water quality. However, cultivating rib mussels in hatcheries presents challenges, as this species is still new to aquaculture, leading to production bottleneck. To address this, our project explored wild larval collection as a method to augment hatchery production. By combining hatchery efforts with wild collection, we aim to meet the demand for large-scale deployments in polluted environments, enhancing the effectiveness of ribbed mussels in bioremediation. Therefore, we were interested in examining scalable and transferable methods for ribbed mussel recruitment in the field and in the laboratory. The field experiments were conducted at 2 sites, Bergen Basin in Jamaica Bay and Flax Pond. We focused on different substrates and predator exclusion techniques to enhance mussel recruitment and survival. Here is an aerial view of the 2 study sites, Bergen Basin and Flax Pond. Bergen Basin is located west of JFK Airport. It is heavily impacted by urbanization and poor water quality due to combined sewer overflows. This makes it an ideal site for studying the bioremediation potential of rib mussels in a challenging environment. In contrast, Flax Pond, a tidal estuary on Long Island's North Shore, provides a more pristine environment. By comparing these 2 sites, we can get insights into how rib mussels perform in different ecological conditions. In the lab, I had intended to replicate the field experiments in a controlled environment by placing the raised larval rib mussels in downwaller tank replicates that had different substrates. However, there were challenges with larval growth, and that delayed the settlement of the larvae. It is still ongoing, but at this point in time, I had to switch to focus on the field trials, which were ongoing. So in our field experiments, we utilized 2 methods, SIPO cages and oyster stacks. SIPO cages are designed for shellfish culture and collect wild spat. These cages can be deployed intertidally and subtidally and allow for easy transfer of recruited mussels to aquaculture setups. Oyster stacks, developed for reef development and coastal resiliency, offer a novel approach. They include screw anchors with copper baffles to reduce predation and can hold different substrates for recruitment studies. Like SIPO cages, they can be deployed in both intertidal and subtidal zones. A key advantage of their removable plates, which facilitate the transfer of recruited mussels. So this is our substrates for the SIPO cages. A is core rope, which is coconut fiber woven into like a net of sorts. B was our second substrate, which contains bees. Bees is a company that makes this like, it almost feels like plastic, but it's actually made out of potato starch. So it biodegrades over time. This one specifically degrades in less than 5 years. So it's perfect for larval recruitment. And the bee substrate had bees, core rope, and core mat. So both the coconut fibers. And then C had the core rope net again, but we had rib mussel shells collected from the site embedded in it. It had 10 specifically. And then for SIPO cages, we had predator exclusion and non-predator exclusion. The predator exclusion had 1 millimeter mesh bags lining the inside. And the concept was that the larvae can get inside the cage and set on the substrates that we put inside, but predators couldn't get in. And then for our oyster stacks, we had 3 different substrates. We had slate, soft bristle with wood, and we had coir again, which is the coconut fiber. And similarly, we had the mesh 1 millimeter predator exclusion bags on half of the substrates, and then on the other half we had non-predator exclusion. So we deployed the SIPO cages and oyster stacks at Bergen Basin on June 17th. The structures were monitored and cleaned every 2 weeks with a total study duration of 8 weeks. After the 8-week deployment period, we collected samples for lab analysis, focusing on the size and abundance of juvenile rib mussels. However, due to weather inconsistencies and scheduling, the oyster stacks are still in the field and will be collected next week. In terms of relative abundance, we observed that the coir and rib mussel shell substrate with predator exclusion had the highest recruitment rate at 25%. However, the coir and the coir plus bees substrate without predator exclusion also showed significant recruitment at 13.79% and 22.22% respectively. This suggests that the substrate type plays a crucial role in larval settlement, but predator exclusion is particularly effective when combined with the conspecifics like rib mussel shell. When we look at the size of recruited mussels, the coir plus rib mussel shell substrate with predator exclusion, which is the mesh bags, also produce the largest average mussels at 9.44 millimeters. In contrast, mussels recruited in unprotected environments without the mesh bags were generally smaller, with the largest mussels measuring 5.7 millimeters in the coir plus rib mussel shell substrate. These results indicate that predator exclusion not only enhances recruitment, but may also allow mussels to grow larger. Our preliminary data suggests that the coir plus rib mussel shell substrate with predator exclusion is the most effective treatment for promoting both recruitment and growth. Interestingly, coir combined with bees also performed well in terms of recruitment, though the size of the mussels was smaller. This could imply that conspecifics like rib mussel shells offer preferable settlement substrate, and predation appears to be a key factor in controlling both recruitment, size, and abundance, as we observed larger and more numerous recruits in potential environments. So in conclusion, our findings indicate that the coir plus rib mussel shell substrate with predator exclusion may be the best approach to capturing wild larvae. This method could be scalable and directly transferable to other restoration projects. Also, we are continuing to collect data and we'll explore whether there's a fall recruitment pulse further to validate our results. Ultimately, this approach could be instrumental to reducing bacterial loadings and impacted waterways like Bergen Basin. I want to thank Matt Scapani, my advisor and mentor. Also my Cornell cooperative team that drove to JFK with me almost all the time, and also picked through coconut fiber with me for like 10 hours. So, and our collaborators at CUNY, Stony Brook, and NYC TDP. References? Thank you. Anyone have questions?
It's definitely time for questions.
Do you have any indication of what the other predator exclusion looked like? Because I know you said you didn't collect the data yet.
Yeah,
We have no idea.
well, yeah, like we only did one of each sample.
Gotcha.
So it's like preliminary.
Yeah, absolutely.
That's tomorrow.
I was curious.
Yeah.
This is not my area, so this might be a dumb question, but I'm just kind of curious whether there's a concern that the predator exclusion will select for individuals that have traits that work really well, like fast growth size, in the absence of predators, but might be maladaptive in a more natural environment with predators.
Yeah, we consider that, but yeah, that's a good question. I'm not really sure, honestly. Yeah.
Do rib mussels only recruit once, and once they are attached, they stay there, or is there some secondary mobility that may occur after they settle?
I know they have a recruitment in like June, and then they have a fall one. That's what we're guessing. And once they set, they set. Yeah.
Any other questions? Okay, thanks a lot. So. Right, okay. Next up, getting ready. His faculty mentor is Brad Peterson. And his internship supervisor is Scott Gerber from Endeavour.
That's working?
Yeah, it works perfect. You're
Okay. So I'm Royce Kang, and my summer internship was with Virginia Institute of Marine Science. And it's the assessment and forecast of marsh habitats through the marsh equilibrium model, or MAM. So internship details, sorry. So where was I? So internship details. So this is part of the Chesapeake Bay National Estuarine Research Reserve in Virginia. So the headquarters are located at the mouth of the York River in southeastern Virginia. And it's part of the National Estuarine Research Reserve System NERS. So it's a network of coastal sites designated for protecting and studying estuarine systems. And my duty for about a month and a half was assisting in field work, but the main beef was using the MEM to record, to move modeling efforts forward for VIMS in addition to field observations and data. So motivation, so wetlands and salt marshes that we address, they're important ecosystems, but the main benefit that I'm focusing here is they're very high carbon sequestration capacity. It's about 3 to 5 times greater than tropical forests are. And this is important in the light of recent climate change and global warming due to excessive greenhouse gases in our atmosphere. And today they are most threatened by accelerated sea level rise, and in the Chesapeake Bay specifically, in the past century, they saw a sea level rise about more than a foot, and they are predicted to rise another 1 to 5 feet over the next century. And the rate itself is faster than the global average, so that's another concern. And the CB near Virginia is engaged in understanding marsh vulnerability to climate change, whether it's natural or anthropogenic stressors in the York River estuary. So study areas, so there are multiple components along the York River, you see there, along the salinity gradient of the river. So my focus was on 2 areas, the 2 downstream ones in the red circles, Goodwin Islands and Catlin Islands. So these 2 are similar habitats in general. They're tidal wetlands on the York River. For the most part, Goodwin salinity is slightly higher than Catlin because it's more downstream. Okay, so for dividing the marsh. So when we were out in the field, it was important to specify different areas of the marsh. So we divided them into low and high marshes. The low marsh is flooded daily, regularly due to tidal activity, relatively lower in elevation, and the dominant vegetation there is spartina alternative flora. And the high marsh, the flooding is more irregular and infrequent because it's more upland and relatively higher elevation. And the 2 dominant vegetation we recorded there from the data provided was Spartina patens and the Stigla spicata. So yeah, it's a nightmare, I know. So this is the model we used, coastal wetland equilibrium model. Everything is a mouthful. So this is just an example, a lot to unpack, but just the main point, the left photo is just an example of the interface of the model where we put in our data. It uses an Excel interface. And the right is the computations page where the outputs are recorded on the 6 graphs you see over there, and in the yellow doodles box using the interface. For us, due to time and data limitations, we had to choose one of the sites that were hardwired into the data you see in the small box here. There are lists of sites that are hardwired in the model. So we chose North Inlet from North Carolina because my internship mentors determined that it was most similar to what we have at the York River. So model outputs, I won't spend too much time on it other than to say that the 3 graphs I circled, inundation time, how much the fraction of the time marshes are flooded or underwater. Carbon sequestration rate, important part because that's what shows actively drawing out the carbon dioxide or excessive greenhouse gases from the atmosphere. And surface elevation to mean water level. That's important because marshes have to stay above water to actively pull out carbon dioxide from the atmosphere. And if they're inundated for too long, the stress can cause them to lose their vegetation, which in turn converts vegetated wetlands to non-vegetated wetlands, such as mudflats, which can hold carbon, but can no longer actively draw carbon dioxide from the atmosphere. And in the doodles, the little red circle there is the end of simulation carbon sequestration rate, averaged over the whole 100-run period of the model runs. And the year of inundation is taken from the computations page in the highlighted column. where the year of inundation will be the first year where the marsh elevation relative to the mean sea level is less than 0. Parameters. So parameters, this is where we input our own field data. The data I received was from August 2018, so that was quite a while ago, but some parameters are more present day, which I'll discuss. Excuse me. So one of the inputs is sea level forecast, and there were 2 scenarios: low sea level rise of 70 centimeters and an intermediate sea level rise of 110. Both were taken from the IPCC 6 assessment report working group 1. And for the intermediate run, 110 centimeter rise, we made an additional run of a single thin layer placement treatment, or TLP, where we apply 14 centimeters of sediment layer to the marsh elevation. So we add the number 14 to the initial marsh elevation for these runs. And starting sea level rise rate was from the Yorktown tidal station, which was the nearest tidal gauge on the sites. And biological literature data was for some parameters we used from the literature are below ground to above ground ratio. These were different for the 3 different species of dominant vegetation, so this was very important. And refractory fraction, this is the lignin concentration in the vegetation tissue. This is also important because it's what adds to the below ground biomass and hence helps the marshes to vertically create or stay above the sea level rise. And again, these were different for the 3 species as well. And field data provided us with the initial marsh elevation, accretion rate, flood frequency, and biomass levels. This is just an example of how the data is collected. Left, this is me measuring the marsh elevation along a straight transect using real-time kinetics. And it compares the elevation to the elevation set at the surface elevation table in the middle photo where my mentor is setting up the surface elevation table or set. This records the elevation of the marsh very deep down, and it measures it in a fixed position over time. And vegetation plot sampling is where we count up the number of species and the density of each species in a 1 square meter plot. The biomass isn't taken from the plot sampling, it's taken from the sediment cores, which is not shown here. And I personally did not do the ownership. So this is a master results table. So for each of the sites, so Goodwin and Catlin, we tried to make it an even split between low and high marshes. And overall, so the first 2 columns show the intermediate sea level rise, the middle 2 columns is the low sea level rise, and the final 2 is the intermediate plus the TLP treatment. And the inputs highlighted in yellow show the marshes that are maintained above sea level, even past the 100-year mark. So carbon sequestration, these are the average sequestration rates grouped by marsh. Left, so the blue bar is intermediate, the orange is the low sea level rise, and the green is the intermediate plus TLP. The results here were interesting because for Goodwin, for the high marshes in Goodwin, there was one data that really shot the average carbon sequestration rate up by quite a bit. So as you can see, there's a pretty big variation of data. So you see, like the last high marsh 5-2, it's quite high compared to the others. It alone is like 200 as opposed to like from 20 to 30 or 40 the rest. And so the average year of inundation, again, grouped by marsh. This is a little bit more even with less variability. And the main point here is that, so the green bars are the TLP treatments, and they do a pretty good job in extending the lifespan of the marsh, although not as much as if sea level rise was greatly reduced, but that's something we can dream about. So data discussion and implications. So most marshes, they do not survive to the 100 years, to the end of the 100 years, which shows that marsh vertical accretion is not keeping up with the sea level rise right now. And the marsh transgression, which is where the marshes actually migrate upland into more upland habitats like forests, that's pretty much going to be the only saving grace for many marshes. And even that could be limited if coastal development limits their transgression to upper habitats. And another important thing is carbon sequestration rates, they just bottom out as soon as almost as soon as when above ground biomass plummets. which makes sense because you no longer have the vegetation to actively withdraw greenhouse gases from. So that was actually an important thing, even more so than sea level rise, which is why sea level rise and inundation time graphs, I put a little bit more emphasis on them more than the carbon sequestration, because what's the use of having a high sequestration rate at the over the 100 years if the marsh is going to be sunk halfway through that? So the Goodwin Island marshes appear to be faring better than Catlet, but as we said, there was that one exceptionally high reading in the carbon sequestration rate for Goodwin. So that's not exactly sure if it really is doing better, but my mentor did note that that the Goodwin marshes may have had higher elevation capital to work with, so they have a little bit more cushioning, so they have a little bit more elevation to work with. So that might be a case. But the biggest find I would say is the thin layer placement. That was just a single placement at the start of this run. So that is shown to be pretty effective in delaying marsh inundation. So future efforts, we could use a more recent version of the model available. And again, apply more site-specific and time-specific data wherever possible. And also conduct a sensitivity analysis on key parameters. And for thin layer placement, okay, it looks effective, but there are some considerations that have to be considered, like costs, limits, and source of sediment, and also the long-term monitoring to make sure that we don't lose these important marshes. So acknowledgements, I would acknowledge SPU mentor, Dr. Bradley Peterson, and my internship mentor at VIM, Scott Werberg, who with Aaron Riley provided me with the necessary field data from 2018, and Dr. William Ray of VIMS, who helped me greatly with the quantitative part of the model, and Dr. Laurie Staver, who is not part of VIMS, but still provided us with the model that we used in the project and additional guidance and some of our own inputs too. Any questions? So, time.
There's time for a question or two.
I mean, if no one has any, can I just explain why I mentioned how global sea level rise average is actually little? So I mentioned offhand that the Chesapeake Bay sea level rise rate is higher than the global average. And a couple of reasons for that is one, intensive local groundwater withdrawal. Second, it is suspected that the weakening of the Gulf Stream is actually causing some of the water to slosh back towards the East Coast, where the Gulf Stream would have taken it, but is weakening, so the water is coming back to the East Coast. And third reason is a post-glacial isostatic rebound, which is where, because there used to be a glacier sitting on where Pennsylvania is right now, but once that glacier was gone from the last ice age, it is kind of, think of like a seesaw. So once the glacier is gone, slowly coming back to where it was. But to us, that's considered a land subsidence for Virginia. So that's why all these factors combined are resulting in a sea level rise that is higher than the global average. Just wanted to touch on that because I don't think I mentioned it in the thing.
Great, that's good. Thank you. Jonathan's faculty mentor is Yang Chen. And his internship supervisor is Shannon Bassett from Melbourne.
Hi, my name is Jonathan, and this is my presentation. So aided science, like if science communication matters. So quick question, what was your first introduction to science? Doesn't matter if it was like a movie, a TV show, book, or how even a random science exhibit, what was your first introduction? How did you first was introduced to science?
Old Bill Nye, the science guy.
That's a good one.
You're old, oh my god.
Yeah. Maybe the why I asked this question because like regardless of how you were introduced, these are all forms of science communication. For the sake presentation, the definition I'm using for science communication is about how scientists are discussed with the people and the relationship between science and society. Because even though like science meant to help people, that relationship isn't always the best. Anyone remember the March for Science a few years ago during a certain president's administration? One of the main concerns was literally about like how science was communicating people and how they either were ignoring it or misinforming or people based on more what is about it. So for my internship, it was kind of like how circulation of how speaking that by those my my faculty. There was Dr. Young Chan who was working couldn't be here today, and my internship supervisor was Shannon Noah. So like maybe even what more maybe what probably just like how marine biology are important in both the environment and economy. They're not the most charismatic animals like animals like sharks or whales, but they're important foundation for many environment for many environment. Sorry, I had some dental work done recently. So during the internship, during my internship, there were framework four main objectives that do was it was update content from the welfare lab, generate media, measure public perception of justification, and also assist with any lab or field work that's going on in the lab. So for my approach, each objective had a different approach. Some of them like objective one involved reviewing like what has already been made first and seeing the strengths and weaknesses, like what doesn't need to be changed, and also like what needs to be updated, because a lot it is a number of overlap like for that project, like why the information is good. It just hadn't been updated. Like some of them when referencing projects we cited information from a few years ago rather than like the most recent up-to-date information which was available on other parts of the web page. Well, there's like a bit of two. It just involved we're taking video and video recordings to help to be used to help generate media. Objective three, it was trying to make meetings with different stakeholders. And objective four, the reason involved is helping of any field or lab work that needed to be done that summer. So for the results, good news and bad news. Good news was 3 of the objectives were complete, either according to plan or even beyond. Bad news is 1 of the objectives we were not able to complete. Objective 3, a lot of the meetings we had scheduled by the summer. Either had to be cancelled or rescheduled to the fall, to the fall. Attempts to try to find alternatives didn't work out. Or for the other 3 objectives, for the other 3 objectives, they worked out well. The priority articles that need to be updated for in objective 1 were successfully updated. For objective 2, we plenty of video. We did take 1,000 photos, 2 blogs and 10 videos. Some of those videos including the Senator of Connecticut giving a speech at the Milford Lab. I'll need to provide the only recording that we have in the lab that was had by myself, and he had recorded each thing that day belonged to local news networks, and to get access to that and use them will require us to pay money, so the fact needed to get them recorded help avoid that issue. And for another one we do, it was pretty updated, and probably that was one of my last and one of my more important the summer was mainly was meant to be used as part of our outreach for the fall open house, and mainly for the show like these are different students that are doing at the Milford Lab, and where we come from. Not all of us were from Connecticut. Many of us came from the US, but we all came here. It happened with it. And yeah, these here are just more photo taken throughout the summer. For the broader significance and impact, I mean, when if we're like that, it was made to keep people informed of work being done in done in the lab, and also providing like it also like just having like providing like not only up to date but like but reliable information that's easy to understand. It helps put credibility, and it with people, and frankly, in the end of the day, like no one knows everything. Like not first off, not everyone's a scientist, and even then, not every scientist is an expert in every scientific field. So regardless if you're part of the scientific community or not, or you're complete a stranger who never picked up any science materials like that, the point was trying to create a content material that's easy accessible to people, but more importantly, easy to understand. Yeah, because first because something that we found doing something I found doing was that social media was very useful to help with science education. Anyone does anyone remember the what fat bear week or the ugliest animal contest? The point was these weren't like official scientific paper or even like the reality. These were just literally social media campaigns meant to help generate interest. Fat bear week and only got people more interested in the bear. They literally got it literally on TikTok alone, hundreds of videos were made. With millions of viewers. That's outreach that you can't buy. That's we get like the guarantee advantage of people's interest is, but it's an natural but who want to know more, but they currently don't know more about it. Also, humans at time found to be very useful. Like in the ugliest animal contest, they're not as famous as like the big cats and the elephants, but some animals just like the blood of the first how of animals who need protections, well otherwise don't usually get that much attention. Like there's an example of last year's Fat Bear Week. They have it every fall, so like there's let us know 2024 one because that's not gonna happen until a few months from now. But this was last year's one on the National Park Service webpage. Science is not that simple. It's it's complicated, and frankly, it can be polarizing some days. Some of the issues are controversial, like deep sea mining and fishery regulations. Depending on who you ask, some people think no fishery regulations are too strict, while others say they're not strict enough. But we need to like some of the work being done, they done out to my intensive one. That was very useful to help build trust and credibility. These were the citations I used to my paper, and these are my citations for the illustrations used in this presentation.
Thanks, Jonathan. We're taking questions for Jonathan.
Yeah, Jonathan, thank you. I really appreciated the way the slides were put together, made it easy to follow for someone who's not an expert in that area. But could you tell me more about how you would anticipate using social media to be more effective in this field?
Hold it, or you can put it down and top. Yeah.
Something we found useful doing that was very looking to see what it was into. That's why we tried to. That's why during one year that we found we want to like with different stakeholders during the summer we because those meetings were it does kind of help like figure out like what kind at Jamaica, I did a more informal version, which really just involved like walking around on and asking people directly. Then actually, and mainly we just like for just like this, just figure like what are people interested in first, like. Since for us, like the most for like most of the work with Bible first, where we wouldn't be talking about like fish or marine mammals. That was so like once figuring that out, what we do have, like what would people for a lot of people like for connect where like where the oyster. Especially like every August they literally have something called oyster fest. I realize maybe we should focus kind of that because oysters are especially Eastern oysters are important to the economy. Economy, but also just like most people they just view them as just the weird thing growing in the side of the harbor and nothing else. So it's like maybe we could like like during the oyster fest this year we brought back we're bringing back the touch stick, and we even have some Eastern oysters for people for people to touch to get a close up look at it. Most of them was so people can see them eat them in new light that they usually don't get the attention that they deserve.
Other questions? Okay, thanks very much for Pompey. Her faculty mentor, Neil.
I can just press the arrows to move the slides. Right?
Yes.
Okay. Hello, everyone. My name is Brianna, and I did my capstone on oyster valve gaping behavior under changing environmental conditions as a proxy for particle removal under advisor and mentor Dr. Nils Wolkenborn, and also in association with the Peconic Estuary Partnership. So currently in the Peconic Bay, there is a shellfish aquaculture lease program in which corporations or private entities may lease 10-acre plots for the purpose of oyster aquaculture. And we know oysters are suspension feeders filtering large amounts of water in a day. And in doing so, particles are removed from the water column and deposited into sediment. And this provides a really great ecosystem service in that it really improves water quality. And this ecosystem service, in coalition with how oyster farming provides jobs to baymen, this presents a really great co-benefit, which is utilized by nutrient credit trading programs in other states, and is something similar to what we want to try to get started on. But the question we really want to ask is this ecosystem service site-specific? Are there certain sites where growth conditions are better or particle removal conditions are better? And in knowing this information, how can we maximize this co-benefit produced from oyster farming? So the objectives we wanted to tackle here were, first, we wanted to investigate a method that we can use to be able to predict the amount of particles being removed through an oyster farming project. And we hypothesized that valve gaping behavior could be used as a proxy for that particle removal. We also wanted to assess water clearance services provided by an oyster aquaculture project, and we also wanted to use the data that we collect to help inform site selection and operation type. And hopefully we can not only apply this information to the Peconic Estuary, but also to similar ecosystems worldwide. So the main star of the show really are these Hall effect sensors, which are attached to our Arduino loggers. And these sensors continuously collected that valve gaping data from the oyster that it was attached to. And how it works is that the sensors generate a voltage difference when the circuit is completed. And a completed circuit consists of a sensor sat on top of one valve of the oyster, and then on the direct opposite end of the other valve of the oyster, a magnet is attached. And as the oyster closes its mouth and opens its mouth, the sensor and the magnet come together and come apart. And that signal is constantly recorded by the Hall effect sensors and the Arduino loggers. So this is our experimental setup. It looks kind of messy, but I promise you it's very organized. I learned a lot about cable management throughout this project. Those are managed. So this is a multi-stressor system we set up at Flex Pond. And through this system, we can control a wide variety of environmental factors that these oysters may encounter in the wild. So we can control temperature, dissolved oxygen, pH, and what we really focused on through this portion of the project, which I'm going to show you, is the temperature stress of 25 degrees Celsius. There were 8 oysters in each bin, a total of 3 bins, and each bin received a constant flow through of water from Flex Pond at about 24 liters per hour. And the first bin here, this was our control bin. They only received water from Flex Pond. So the only food they were receiving was whatever is in Flex Pond at that time. And then this second bin here, this is our bloom bin, where we were able to replicate a phytoplankton bloom through an application of a pure algae culture, SoMAS, at a rate of 76.8 milliliters per hour. And then our last bin here is our intermediate. It's supposed to be somewhere in the middle of the control and the bloom bin, but we weren't able to establish a consistent flow rate of that algae. There was some faulty tubing, so we're not gonna present anything about that in this presentation. But in each bin, the cell density was assessed by flow cytometry at the beginning and end of each experiment, so that we know how much algae was experienced by those oysters. And then each oyster was placed in a petri dish with mesh walls so that we can collect those feces and pseudofeces, but at the same time allow food to reach them through the mesh. And those particles produced by the oysters were used as a proxy for clearance. So this is how I collected the particles released by the oysters. And then after we collected those particles, we put the water samples through a vacuum filtration apparatus where they were filtered through a 1 micron filter, and then we took that, dried it, and got the dry weight of the particles. So this is the raw data put into a graph. This row here is one single oyster, oyster R43, from the control bin, the no food addition bin, and this row here is one oyster, oyster Y45, from the bloom bin with high food addition. And how you interpret this graph is that a high millivoltage reading means that the oyster is closed. That's fully closed all throughout here. And then a low millivoltage reading means that the oyster is open. So what you're seeing here is the oyster is throughout that 1 month, right? And then, so looking at this, you can already see a difference between the control oyster and the bloom oyster, and that I can count these. There's 32 total opening events within that 1 month. But in the bloom, I'm not even gonna count that because there's too much, they're overlapping, there's too many going on. So how do we quantify and analyze this data? Well, we have an R script and we tell the script that anything above this line means that the oyster is closed. Anything above there is closed, anything below here, the oyster is open to some degree. And then the R script uses that information in the data to create several different matrices, and 3 of which we found particularly useful were the percent of time open, the duration of those openings, and the amount of openings per day. And then from that information, we can create graphs such as these, which tell us really valuable information, like here, in the no food addition tank, they were open for about 25% of the time on average. And then in the bloom tank, they were open for also about 25% of time on average. But on average, the bloom oysters created about 3 times as many particles as the control oysters. So they were open for about the same amount of time, but created way different amount of particles. And then when you graph them on the individual level, you see a correlation between the percent of time open and particles per day. But to get that correlation, you need to consider the algae concentration in that tank. So for the high algae addition oysters, we see an 85% correlation here. And then for the control oysters, we see 87%. So that means that with 85% predictability confidence, we can predict the amount of particles created per day using the percent of time open and considering the algae concentration. but it's not quite 100%. How do we get closer to 100%? So we hypothesized that the correlation between gaping and particles produced will improve further when you consider the amount of partial closures associated with feces and pseudofeces expulsion. And so there you see the 48-hour data, again, from the previous slides. And then you zoom in to one opening event, one gaping event, and then you get these two graphs. These are just about 4 hours long, different time periods, but just about 4 hours long for each period. And you already see that there's a lot more going on in the oyster in the bloom tank than there is in the control tank. These bumps that you see here, those are partial closures. So the oyster is open, but they're just like this, partially. And we think those are associated with feces and pseudofeces expulsions. And there's a lot more of those partial closures happening in the oyster in the bloom tank. And once we get the frequency of those partial closures, maybe we can increase that correlation from 85% to maybe close to 100, maybe not 100, but close. So in summary, we discovered that oysters only spend about 25% of time open. And that percent open is indeed a good proxy of particle deposition if you're given the algae cell density. And that if we take the data that we found and consider same same conditions as what we had in the laboratory, one oyster bagging Porites with a maximum of 200 individuals may remove upwards of 500 grams of particles from the water column per bloom period. And we also found that partial closures may serve as a proxy of particle deposition for any algae cell density. So we need to work on categorizing that. So the next steps we're working on is we want to create an improved data analysis to get automated detection of those partial closures. We also want to do field deployments at Flax Pond where we can get valve gaping behavior in response to other environmental conditions like current velocities, temperature, salinity, DO, etc. We also want to deploy floating and bottom cages so we can understand how we can maximize that co-benefit further from those floating and bottom cages and employ that in an oyster aquaculture operation. So I would like to thank Dr. Nils Oldenborn, my awesome advisor. He dedicated a lot of time to helping me throughout this whole entire project from the beginning of the experimental procedure to the paper writing to everything. I really appreciate it. Dr. Bassem Alam for letting us use this flow cytometer. I can't believe he let me use that alone. Ian for writing that R script and helping us use the Hall effect sensors. Dimitrios for providing us with those pure algae cultures. Thank you. And Greg for giving us those oysters. And Joyce for offering this really great opportunity for me. And also the poor oysters that dedicated their lives to this project. Thank you.
We have some time for questions. Got a question back there?
Yeah, that's a nice presentation. Most people are usually popping champagne bottles when they get an R squared like 0.35. So I'm trying to get a 1. I was just curious, so that you had a 25% of the time open. And one of the big sales pitches for a lot of oyster restorations is that they can clear 50 gallons a day as an individual. Do you think that there may be overestimating that just a bit? Or I don't know, they assume that it's what the prevalence of filtering, but do you think that's a possibility?
I mean, I definitely think it's a possibility. That even in the bloom tanks, they didn't get extremely dirty. So they're definitely filtering a lot per day. Especially, I think like later on, we're gonna try to get some clearance rates and see exactly how much they're filtering in that day. So maybe we can verify that. But I think it's definitely possible. The oysters are constantly trying to eat. Even in the control, they were just open, just filtering through water, just trying to get anything they can get. So yeah, I think it's possible.
Other questions? There's not very good questions. All right, thanks
Thank you.
very much.
Did not.
Daniela's mentor, Leslie Thorne. Leslie?
She couldn't make it today.
Oh, okay. And internship advisor, Ellie Siwak, who was here, still is here from R.I.S.E. Thank you. Okay.
Can you guys hear me? Okay, this is running. Okay, so my project is using aerial surveys to estimate abundance of pinnipeds at haul-out sites and analyzing pinniped strandings in the New York Bight. I did my internship through the Atlantic Marine Conservation Society. I initially came on as a volunteer and then ended up doing my internship with them. So I'll just briefly go through some of the things I did at my internship. I know Cassidy covered it pretty well earlier. I was a part of the sea turtle rehabilitation. There were 20 endangered hence Ridley sea turtles under our care. 16 of them were released in April in Georgia, and the other 4 were released later in July. There, I helped with feeding the turtles, cleaning their tanks, taking water quality, and collecting morphometric data on them, tracking their growth, and helping with their treatments. And the sea turtle release, this was extremely rewarding to see these turtles go back into the wild after all the hard work that went into them, and seeing like the changes in the turtles since they first came there. Some of the turtles, they weren't moving as much, they didn't eat that much, and then by the end, all the turtles were very healthy. Also, seal cruises. Here, I helped take pictures of the seals on the cruises, help talk to people on the boat, and collected data. Here's some pictures from the seal cruises. These are harbor seals. And education and outreach events. So this included many beach cleanups where I helped collect marine debris, document the different types of debris, go to different events where I talked to people about the mission of Atlantic Marine Conservation Society, and also teaching necropsies where students got to learn what goes into a necropsy, the different data that we collect, the different kinds of samples, and what we look for. And the stranding response, I'm sorry, these pictures are a little rough. I got to help respond to multiple species of dolphin, 3 species of seal, harbor, harp, and gray seals, and help collect level A data on them, and also participate in the necropsies and help take samples. And my research questions for my project were which pinniped species have stranded in New York from 2017 to June 2024, and what is the frequency of stranding for each species? Where are pinnipeds stranding throughout New York State, and what are the hotspots? What are the average straight lengths of stranded pinnipeds, and how does this compare to average lengths of adults of each species? What is the abundance of gray and harbor seals at surveyed haul-out sites on January 8th, 2024 and April 23rd, 2024, and how does the abundance vary between survey dates? And how can these findings be applied to future research and improve conservation efforts of pinnipeds? So that my materials and methods for the pinniped stranding analysis, I use the Atlantic Marine Conservation Society Master Stranding Database. I sorted through the level A data and I created separate data sets that I was able to find average straight length and create maps using ArcGIS Pro. And for pinniped abundance, I used the AMC's aerial surveys that were conducted by Chief Scientist Robert D. Giovanni that took place on January 8th, 2024 and April 23rd, 2024. All these surveys took place within 2 hours before or after low tide. And I also utilized the AMC's master aerial survey database, which is where the seal counts from the aerial surveys get entered. And from there, I would identify the species of pinnipeds in the images and count the seals. For my pinniped stranding analysis of 536 total pinniped strandings from 2017 to 2024, 263 were gray seals, 104 were harbor seals, 134 were harp seals. There was 1 hooded seal, and 34 seals were unknown or they couldn't be identified. And this year, from January 2024 to June 2024, there was 29 gray seals, 7 harbor seals, 16 harp seals, and 3 seals were unidentified. And this year, harp seals accounted for 29.1% of pinniped strandings from January to June. They are Arctic seals. And this was pretty interesting to me because I found a study that years with light sea ice cover have increased strandings of harp seals. So I think this is something that is interesting as we're going to see more years with decreased ice cover because of climate change. And this was the map I created of the pinniped strandings from 2017 to June 2024. You can see there's a higher stranding density in West Hampton, Montauk, and Fire Island for seals. And the average straight lengths. This is the total average from 2017 to June 2024, so it doesn't represent the change in the lengths of the seals since they've been stranding. But all of the seals, the overall average is below average for each species. So this suggests that these seals are yearlings or subadults. And that same study I mentioned before found that yearling harp seals are more likely to strand than adults. This has to do with years of light sea ice cover, because when there's light sea ice, the seals are forced into the water earlier, and they may be not as ready to enter the water and begin foraging on their own. And it can lead to decreased body condition and cause them to fail to thrive when they come down here, and which can cause them to strand more frequently. Now for aerial surveys. This is a map that was provided by the Atlantic Marine Conservation Society of harbor and gray seal haulout sites in southern New England and New York. And what I did with these images from those two aerial surveys was I counted seals in each image. And first I had to identify the different seals that were in the images. The two seals that we see hauling out in New York are gray seals and harbor seals. We don't really see hooded seals haul out. They're considered vagrants in New England waters, and little is known about them here. And for harp seals, they rarely haul out on land, so we don't see them. So on the left are gray seals. They're sexually dimorphic. You can tell them apart from harbor seals because they have a longer, more horse-like face, and the males will have a darker coloring with light spots, and the females will have lighter coloring with dark spots. And then on the right are harbor seals. They are more like rounder, they're smaller than gray seals, and they have a shorter, more dog-like face. And what I would do is when I was counting haul-out sites, I would look at a reference photo of the entire site that was taken during the aerial survey, and then every image that I counted from the site, I would make sure accounted for the entire haul-out site, and I would mark it on there to make sure that I counted all the seals in the area. That's Little Gull Island on the left, and on the right, you can see Fort Tyler. At the top, that is the entire haul-out site, and then on the bottom is an example of one of the images I would have counted that would have made up the rest of the haul-out site. And the results of the seal counts from these two aerial surveys from all of the surveyed haul-out sites on January 8th, there was a total of 278 seals seen, and 129 of them were gray seals, 134 were harbor seals, and 15 were in the water. On April 23rd, 2024, there was a total of 1,080 seals counted. 860 were gray seals, and 207 were harbor seals, and 13 were in the water. We can see significantly more seals in April than there were in January. And this is the breakdown of seals per site. So you can see which sites had the highest amount of seals and which kind of seals. In January, there was the most seals at Little Gull Island, and they were gray seals. And then in April, you can see that the most amount of seals was at Block Island, and these were also gray seals. And back to changes in seal abundance. So since the Marine Mammal Protection Act in 1972, pinniped populations have been growing in the United States. Gray seals were considered extinct in this region prior to 1958, and harbor seal populations have quintupled in size by 1999 since the passage of this act. And related studies in New Jersey show that harbor seal abundance has increased significantly in the last 15 years, which was in the Great Bay of New Jersey. It was a very similar study of counting seals and haul-out sites there. And we're seeing large numbers of seals hauling out in New York, which is reflecting their increasing populations and could suggest that they're utilizing the southern extent of their range more. Some threats that these animals face is fishery interactions. So gray seals experience the highest level of annual incidental bycatch in the New England St. Gillnet fishery compared to all other marine mammal species in the United States. They also are threatened by entanglements. You can see on the right, those are 2 gray seals with signs of entanglement that were actually from the April 23rd, 2024 aerial survey, and that's at the north point of Garner's Island. Climate change is also a threat to seals, as I mentioned before, decreasing sea ice and harp seals is a threat to them. It can lead to increased strandings and pollution, disease, and coastal development, as well as vessel strike are threats that these animals are facing. For future research, I think that the current seal research that's being done by AMCs is really important in understanding the growing populations of these animals and how they're utilizing their environment. Also, counts from aerial surveys can be used to estimate seal abundance in conjunction with data from tag seals, because you can use that data from tag seals to account for the amount of seals in the water and adjust that number. Also, I think changes in seal abundance at specific haul-out sites that have been surveyed by AMCs are future research that can be done, because we can see the change in those haul-out sites over time and how the seals are utilizing it. Also, the impact of increasing gray seals at traditional harbor seal haul-out sites and how these species are interacting with each other, because they have very similar diets. Also, more research on harp seals, understanding why these animals strand here and their diets and the factors that can lead them to being in decreased body condition when they're stranding. Also, more research on climate change and how this is going to further affect these seals. And also continued research on fishery interactions, just continuing to monitor threats to seals in the area. And also the impacts of seals on local fisheries as well, because fishermen have always considered seals to be competitors to them. So I think it's just also important to understand how they are affecting the environment as well. And the continued AMC stranding investigations are extremely important in understanding why these seals are stranding and recognizing trends or new changes in their strandings. I think that's extremely important. And I just wanted to say thank you to everyone at Atlantic Marine Conservation Society for helping me with my project and for their expertise in the area. And thank you to Dr. Thorne for helping me with my project. Does anyone have any questions?
Great, we have 3 minutes for questions.
While the policies, did you notice a lot of seals with parasites or stomach worms? Cause I remember when I did a few crossings, I remember I had a few seals with that, and I thought that was pretty strange. A lot of them had stomach worms. Do you think there was a lot of those, and you know why?
I did notice that a lot of them did have parasites. I'm not exactly sure why or if that is like a threat to them. I know that a lot of wild animals live with pretty heavy parasite loads, but I did notice that they did have parasites in their stomachs.
So say you find an entangled seal in an aerial survey, what would be done? Would they like calling one for that seal, or just set a date?
Well, in this case, by the time I got to these photos, it was months later. It would be pretty hard to find that seal. That seal's probably migrated north by now.
Yeah.
But I'm sure there are groups that could handle that if it was somewhere also more accessible than where that seal was, and if they caught it right away.
Another question?
Is there excuse me, is there a particular time of year that would be ideal for monitoring seals on these particular beaches?
Yeah, that would be from, I believe, December to May is when we see the most seals here.
And do you expect that there would be could you compare a December survey to a February survey, or would it, are those apples to oranges in terms of the population dynamics?
I feel like it would probably depend on a lot of factors, probably weather, food abundance, but I know that from the surveys I've looked at, there's more seals later in the spring than there are like earlier in the winter.
Okay, that's all the time we have.
One more question.
Can you quick?
Quickly, just,
Okay.
I saw a gray seal in July over in Montauk. Would that be normal?
I do believe we're noticing seals staying longer, but that's also something you could, report in that you notice a seal. You could call the stranding hotline and inform them that there's a seal there, maybe there could be something wrong with the seal. But we are seeing that seals are staying longer and some seals are actually staying all year, just not in numbers like those images.
Okay,
Um,
great.
I also put pamphlets in the back, brochures for AMCs if anyone is interested in learning more or getting involved. Or if you wanna ask me questions at the end.
Great, thanks a lot. Richard. Yeah, the cheering squad with her. And her faculty mentor is Joe Warren. And her internship supervisor is Thomas Gillen. Is Thomas here? ASA Analytics, no. Here we go.
Can you guys hear me okay?
Yeah, that's good.
Okay. Hi, I'm Emily Wickers, and today I'm gonna be talking about larval fish and fish egg entrainment at Northport Power Station in New York, which is right here on Long Island. Just a quick introduction for anyone who isn't familiar with entrainment. Essentially, entrainment is when small organisms pass through a water cooling system that is used typically with industrial sites like power plants, and then are discharged back into their original water body. This is a really big issue for those small organisms and their populations because those organisms, when they're sucked in there, they can be sliced open by the pumps that are used to draw in water, or they can be hit with really intense temperature changes, which can shock them and cause them to die. This is something that affects a lot of smaller organisms, but regulations are specifically geared towards shellfish and fish. So we're going to be talking about ichthyoplankton today. And that is essentially just early life stages of fish. This is when they are eggs, when they are yolk sac larvae and post yolk sac larvae. This is just a quick chart of what that looks like. When they first hatch, they have the yolk still attached to them. They're completely translucent, and then as they grow, they lose the yolk. They become more pigmented. These are just some pictures that I took while I was on the job of some of these post yolk sac larvae. They were easier to photograph because they're not translucent. The regulations that are based around this, from the EPA and the New York State DEC, they essentially expect that every single power plant that uses cooling systems like this will kill 100% of the fish that are entrained. And they do individual studies on each of these power plants to prove that the mortality is less than that. When it comes to Northport Power Station specifically, this is located right on the north shore of Long Island. It's facing the Long Island Sound, so it uses cooling water from the Long Island Sound. It does this through the intake canal that's on the side, and then it passes through the power plant and is discharged into the discharge lagoon, which eventually is flushed back out into the Long Island Sound. This is just a once through cooling sort of mechanism. They don't reuse any of the water that they have. It just goes right back into the sound. And in 2016, the Northport Power Station came up with a new monitoring plan with the DEC so they could implement new screens with fish protection so that they could keep more of these fish larvae and eggs out of the power plant. And they kind of diverted from this plan a little bit because they decided to use some screens that hadn't been tested before by the EPA or by the DEC. So they enlisted our help at ASA Analysis and Communication to go and complete a 2-year study to see what the survival was for the fish that went through entrainment here. I'm not gonna go too in depth with the methods of how we did this. I wasn't really allowed to take any pictures of the power plant because it's a federally mandated facility. So I just took some pictures from where we did most of our work in our trailer. We used a barrel sampler and a jib crane to sample from the shores and both the intake and discharge sites. The intake sort of acted as our control in this study. We filtered all of the water that we pumped through this barrel sampler through 335 micron mesh, and that just kind of kept out all of the bigger organisms. And we sorted out from there, we emptied out the sample into a smaller box that was sorted by sorting technicians. They took out all of the dead and larvae and eggs. These are not really important for our study. All of the dead larvae and eggs that we found. What is important is basically that we found, we took all of the live and stunned larvae and all of the live eggs, and we kept them and held them to see how long they would survive. And then from there, the latent technicians, that's what I did. We checked on them every 6, 12, 24, and 48 hours after the sample collection. And then after 48 hours, we put all of the larvae and eggs into pill bottles with preservative to be sent to another lab to be identified. So our results from this study, at the top here, I have the intake, which is our control, and at the bottom I have the discharge. Initially, we found a lot more living organisms in the intake than the discharge, which is to be expected. A lot of times, sometimes we can't find a lot of the larvae and eggs that go through to the discharge because they are just so destroyed by entrainment that they are completely unrecognizable. We did find a good amount of living organisms in the discharge. After about 48 hours, the eggs that were alive, about 50% hatched for our control and about 22% hatched from the discharge. So there's a bit of a discrepancy there. And then when it comes to survival, this does not take into account the hatched eggs. We see that the control survived a little bit less than the discharge. This is very different from the first year of this study, which showed that the control, they survived a lot more. So there could be a lot of reasons why this happened. We don't know 100% because the data is pretty fresh. We haven't really analyzed it as much as the previous study. So I don't know, this is just, it's very odd to see this. The previous study also did not take into account the hatched eggs for the last final numbers for the survival. But further, there was kind of a, we really kind of expected to go at like in May and June, the survivability is pretty much steady, and then it drops off towards late July because there's like low DO levels. This is the intake and this is the discharge. The intake the survivability just steadily decreased, and initially it was a lot higher than in the discharge in May, and then it just completely decreased. And this could be because of weather changes. It rained a lot less this year than last year. It could be because of DO levels. But also another big issue was the fact that we had a big hole in our data because a lot of the eggs would go missing. This could possibly be because of technician error sometimes, but it also could be due to the fact that eggs can degrade really quickly in between these checks. So they would die and we would not really be able to know for sure if it was because of some sort of error or because of that dying. So there's like this big hole in the data that we can't really know for sure what the situation is for it. So like I said before, this is very different from the previous study. However, we did see that Northport demonstrated a mortality rate of less than 100%. So that's a really great thing. They're meeting what the requirements are. And we also saw that the discharge mortality was similar and occasionally better than the intake. So that would essentially suggest that the discharged ichthyoplankton survived about the same as they would naturally after going through entrainment. However, like I said, this is very different and from the previous study, and the previous study showed that the intake, those survived a lot more than the discharge. So I guess it just kind of varies year from year depending on other external forces, but we can't really know for sure until we analyze the data more. So although we see that they are meeting the regulations and that the numbers are looking pretty decent, it's still pretty bad that there are so many that are found dead already in the samples. And so an important part of really just trying to mitigate those issues is by finding a good balance between ecology and engineering solutions. And this is something that's really important because we need to kind of preserve industry to an extent because it's important for a healthy economy, but we also need to preserve the environment in this while still pursuing industry. And a good example, I'm going to give just a couple of examples of how this works and how it doesn't work. An example of an industrial site that did take ecology kind of into consideration is from Diablo Canyon Nuclear Power Plant that I looked into. They did so many ecological surveys on so many different areas on the site that they were going to be building on before construction. So they really understood the natural patterns, and when they were building it, they kept those things in mind so that they wouldn't be disrupting the ecosystem as much as they would have if they had not done these ecological surveys and these environmental impact statements. An example of when this doesn't really work out is with Belo Monte Dam in Brazil, which did not really have any ecological surveys or environmental impact assessments. They did not really communicate with the local area. So when they built the dam, it caused so much flooding, it caused displacement of the people living around, it caused food insecurity and a decrease in the wildlife populations. And this is because the very nature of the dam really just changed the entire river that it was located in. And this is something that we should be avoiding when we are developing industry and working with ecologists. This can really be tied back into entrainment by showing that we could be doing better by taking a lot of ecological surveys before building power plants, and so that we can really understand what kind of barriers and screens could be put into place specific to whatever kind of organisms are living around there. And this can also be done to already existing plants in order to better assess how we can mitigate these effects. So in conclusion, we were able to do it. We were able to meet the regulations that we needed to make. However, this could be better. Regulations could be a bit tighter, and we could be able to create better change with finding a better balance with all these ecological surveys and environmental impact statements. The slide looks a little bit crazy right now. It's not supposed to look like this. But thank you so much to my mentors, Dr. Joseph Warren and Thomas Gillen. And thank you so much to ASA Analysis and Communication for this opportunity.
Okay, we do have time for questions.
Yes.
What do you think would be a reasonable mortality percentage that they can aspire to reach? Or that restrictions could, so right now it's at, it has to be below 100%.
Yeah.
What could it be below 90%? Could they enforce that?
I mean, I guess I think that it should depend, it should depend more on what the mortality percentage naturally is, and it should just depend on, it should just vary area from area to area, and that we could make a policy based off of what those, how well they naturally survive in comparison to, how well they survive after going through entrainment.
Do you think the restriction for Northport could be lower, or do you think that's?
I think that the restriction, or I think it could be higher. I mean, the screens that they use right now are not supposed to be for protecting fish. They're supposed to be for keeping out debris. So I think that there definitely could be more development on that side.
Yeah.
I was wondering about, so most of the mortality probably occurs with this mechanical injury and stuff like that. How much is the temperature increase during the cooling, and could that also play a role in explaining mortality, or is the temperature difference so little?
The temperature difference is really a big part of why a lot of them die. because it's about a 10, well, when we were sampling on the outside of like where the discharge is, it's probably cooled off a bit since then, but it was about 10 degrees Celsius, the difference between each side. So it was a pretty big difference. So yeah, I do think that that plays a big role in why they die.
Any other questions? Okay. Well, thank you very much. Well, I hope everyone has enjoyed today. It's, to me, it's an incredible lineup, right? I mean, we've heard from people working hyperlocally to places around the globe, from organisms big and very small, and high tech to, low tech. It's always impressive to me to see the diversity of projects, the diversity of ideas that students are pursuing here and what they come out with. So first I'd like all of us to give the graduating students a big round of applause. Very well done. Whoops, sorry, that's my alarm telling me it's time for that. And I also would like to thank the members of the MCP program, the steering committee, and also all of the internship advisors and the mentors for all the students. Round of applause. And of course, Caitlin, this is the director. And to the friends and family who showed up to be here for their friends. So, thank you.